Right.
சார் குட் மார்னிங் சார். வந்துடலாம் தான் நினைக்கிறேன் சார். அந்த இதை கிளிக் பண்ணால் போதும் சார், நீங்கள் ஜாயின் பண்ணிடுவீங்க சார். நீங்கள் வெளியே ட்ராவலில் இருக்கீங்களா சார்? சரி சரி. இருக்கேன் இருக்கேன் சார். ஒரு இப்போ கரெக்டாக ஒரு 10:05 அந்த மாதிரிக்கு வரீங்களா சார்? குட் மார்னிங் சங்கர் சார். பேசுவீங்களா சார்? வந்துடா அவங்களாமான்னு கேட்டாரு. அவங்க ரெடியாக தான் இருப்பாங்க. 10 ஆயிடுச்சா? ரொம்ப 55 சார். புடி இது ஃபாஸ்ட்டாக போகுது. ரொம்ப 55. சரி. So good morning, Sankar sir. So good morning, Jayalakshmi ma'am. We will start in 5 minutes, sir.
Okay, sir. Thank you.
மேம் கேமரா கொஞ்சம் கீழே வைங்க.
Oh,
ஆ
okay.
கண்ணாடி மட்டும் தெரியுது.
சரி சரி சரி. வயசாயிடுச்சுல போடாம பார்க்க முடியல.
அப்படி சொல்ல வரல, தான் தெரிஞ்சது காசு.
கிளார் அடிக்குது.
So hi Manju sir. Good morning. we have a lecture from Dr. Rajendran, and he's giving a lecture this afternoon on the applications of facts on flow cytometry. And Professor Sanger would be giving a lecture tomorrow on the applications of flow cytometry on cardiac diseases. Okay, so these are so I welcome Dr. Sanger, Dr. Rajendran, Dr. Jayalakshmi, and the faculty members of School of Biological Sciences, School of Biotechnology, Maria Kamaraj University, and all the participants, not only from School of Biological Sciences, Maria Kamaraj University, and also from all the institutions across the country. So we have the participants. I think again, I would like to thank Dr. Jayalakshmi, because Dr. Jayalakshmi has sent the invitation for so many students in the last. I think she says she has started only last afternoon, and she has sent a mail to more than 1,000 faculty members and students. And we have the registration of about 500 participants. What is that? Okay, we have 500 participants across the nation, and it's a wonderful platform. So I welcome our Honorable Vice Chancellor of Madurai Kamaraj University for giving this, for giving the permission, and also making this event as a very simple. And so thank you, sir. Thanks for your time and thanks for your participation. And just before starting, I would like to share, my experience. While we start these kind of workshops and conferences, it would be a tiring effort normally. Normally in the school, the students would be involved in decorating the rooms for the whole last evening, and then we faculty members would be working on various aspects of the seminar, and then inviting the vice chancellor and then waiting for the vice chancellor. But our vice chancellor is very simple right now, and he has just joined the meeting from his home. And this is one of the possible advantages of this kind of emergency. Actually, this COVID-19 situation is very bad, pathetic, and nobody likes it. But at the same time, for the academicians, certain things has become little bit easier. Organizing this kind of meeting is easier. And then in this program, 450 students across the country, they are participating, and that is a very good learning opportunity. And thanks for all the participants for turning in, and then your involvement and participation. It's wonderful. I'm sure that in the next 2 days you would be able to learn the principles of flow cytometry and the possible applications in your research, whatever it is, the basic life science, biomedical science, or biotechnology, or any industrial related research. This is one of the very basic platforms. It would be useful. This is likewise, we have planned a series of lectures from Madurai Kamaraj University on various platforms. We have only 5 platforms. One is NGS. I'm talking about general platforms in life science. NGS. Another one is mass spec based LCMS, GCMS based platforms, and another one is microscopy based platforms like fluorescent microscopy, confocal, and another one is this flow cytometry for various analysis of the cells. These are the major platforms which I could think of in life science. And so we cover major part, and then it would be very. I'm sure that these 2-day lectures and this technical sessions would be very much useful, and you would be really become a master in in understanding the principles and in utilizing the facts methods for your. So again, I would like to welcome you all to the event. So now I request our Vice Chancellor to give the presidential address. So இந்த ஸ்க்ரீனில் இல்லையே. Actually, so Sankar sir, can you just give a few words about the conference, and then the Vice Chancellor is not here. I request Sankar sir, our former Vice Chancellor, Head Department of Genetic Engineering, and also active researcher in cardiovascular diseases. He's going to give a talk tomorrow on the applications of facts on cardiovascular diseases. So just few words to the student, the participants and the students.
Very interesting. Good morning to one and all. So thank you very much, Dr. Kumlesan, for this opportunity to just talk few words about this conference. Actually, so this conference, like generally most of the people there, and another thing is in this pandemic situation, most of the people they are conducting webinars for the most of the students, especially the student and research scholars community. But as usual, Madurai Kamaraj universities have pioneered in hands-on training to the students, and we started this seminar, especially Dr. Kumaresan and his team in from School of Biological Sciences. So they started this application-oriented webinar, because actually now in methods, even say for example in this pandemic situation, now most of the people, like the gold standard method is to detect this COVID-19 virus is real-time PCR. So the technology is very much important, because we cannot use any other methods, simple methods or biochemical methods or any other methods to detect these viruses. So the advanced technology is very important, and actually we want to disseminate whatever the knowledge we have acquired and flow cytometer then in various applications. So we want to disseminate this knowledge to the student community. So that is the purpose. And Dr. Kumaresan, Dr. Rajendran from School of Biological Sciences, and the whole faculty, my fellow faculty members from School of Biological Sciences, so they have started this very good webinar on flow cytometry in various applications. Of course, you have read or you have gone through so many research articles, but these people, they are going to talk about their the problems they faced and how they troubleshoot while doing this, because that is very important. So they're going to talk about all those things, and especially from the Beckman Coulter, they are going to talk about the basics of flow cytometry, and like the various fields where you can use this flow cytometry and its applications. Because knowing the very basics is very much important than knowing the other like application, because that is very the basics is very important. Then my colleagues, they are going to, including myself, tomorrow we are going to talk about how you can apply this flow cytometry facilities. And also one more thing is, like from this webinar, we are just disclosing that School of Biological Sciences is having a very good flow cytometry, and if you want, you can make use of them for your research if it is needed. Uh, so, uh, so I hope all the students you'll enjoy this one. And another thing is, please ask as many questions as possible to clear, especially to the Beckman Coulter technical support people. So they are going to give you a very clear idea about the basics and what all the facilities they have and different kinds of machine and different applications and all those things. So try to understand all those things as much as possible, or I would say try to exploit this facility or this information from those people, and then from all other speakers. So they'll, because so if you ask more question, then that'll you will get more information about, because they will prepare based on what they have, just they've come up with some material to disseminate to the students. But if you ask more questions, then it would be really good to get to know more about the flow cytometry and how can use it and what are the problems they'll face and all those things. Even because even Rajendran, he is going to talk about the application of flow cytometry in microbiology. So again, that is very important field. So we can use it. So you can talk more, or you can ask more questions to get to know about the techniques. And I wish all of you will have a very nice time and a very good knowledge about flow cytometry from this webinar today and tomorrow. Thank you.
Thank you, sir. Thank you very much. So, Dr. Rajendran, few words, please.
Hello. Sir, can you hear me?
Yes.
Yeah, good morning to all. So, at the outset, I thank Dr. Kumaresan for giving me this opportunity and for organizing this kind of workshop to our students and participants. Also, I sincerely thank our Honorable Vice Chancellor for encouraging this sort of activities in our school and our university. So this workshop will be like a hands-on experience, of course, not in the lab, but in a video conference meeting. So we have expert from Beckman Coulter and 3 or 4 of our colleagues, senior professors and our colleagues are presenting their experiences in facts and their achievements. And I request all the participants to get more information and more expertise from these participants. So thank you.
Thank you, Dr. Rajendran. So now, actually this event was planned for the research students and MSc students of School of Biological Sciences. And Dr. Jayalakshmi has actually yesterday suggested that, we can reach maybe 1,000 students outside. And now we have more than 500 students. That's only because of Dr. Jayalakshmi. So I request Dr. Jayalakshmi to share a few words before we start the first lecture.
Okay. Thank you, sir, for this opportunity and invitation to present my experiences by using our fax machine. And also, yeah, a very good morning to one and all. Those who have joined us for this webinar, as it is already discussed, it is really a nice platform to reach not of much travel, a comfortable place, and we can learn more rather than spending more of time on travel or wasting so much of our energy on other resources. If we have a commitment to learn, this is a nice platform. And so I wish all of you can make use of this platform to learn. Yes. So as it is already mentioned that we are having the company person with us who will be explaining more of the about the instruments, and really it will be at many times I have noticed that when we are the company person is presenting as they want to reach their product, they put maximum effort to make it simple and to reach the public. So it will be really useful to everyone. You can benefit, and also they will be having sound knowledge on the instrument. So any questions or any clarifications can be made to them, and we other faculty members are going to share our experiences on our respective field, what we have done in the past few years using the fax machine, and how we came out with successful publication. And this is really a high-end equipment. And when we are using high-end equipments, the sample size is what is going to be required is going to be less. When we go for other lower techniques, we need more and more samples. But here, when we are using such kind of techniques, the sample size is they expect even we are able to get a good publication with lower number of samples, because getting clinical samples, and that is sometimes really a tough thing for us academicians, being getting a sample from a hospital and working on it. So we will be sharing our experiences. Yeah, so thank you everyone for joining. Let us make it an academic fruitful event. Thank you everyone. Thank you.
Yeah, so let us start the presentation, So sorry, there is a So good morning again. So now this looks like a fantastic platform. Very good morning to everyone again. And the students from School of Biological Sciences, School of Biotechnology, Marie Kamraj University, and other institutes, faculty members from other places. Hope this event would be very useful to you. And let us, I will give an overview of the basic principles and applications of facts, because most of the things are going to be dealt by Mr. Joseph Muhin from Beckman Coulter. I will just highlight maybe for next 15 to 20 minutes about the basic applications, and specifically in the area of mammalian biology or in human cell biology based applications, and in basic biological research. So you know that analyzing the cells is very important, and that is mainly being done with the cell surface markers actually. So that's being done with cell surface markers. And as I said some time back, there are 5 major platforms in life science research. One is NGS based platforms. You just sequence RNA, DNA, whole genome, whole RNA. NGS is the platform right now. And another one is for focused applications, PCR, either PCR, RT-PCR. It's for gene expression, and then any kind of amplification of DNA, and then SNP analysis, and then for some diagnostic techniques, diagnostics problems, RT-PCR is used. Another one is mass spec. You may be analyzing the proteins, or you can analyze the metabolites by GCMS or LCMS based methods. That is including the methods like MALDI-TOF and everything. So mass spec is another major platform in life science research. And the fourth one is fluorescent microscopy. In fluorescent microscopy, you'll be visualizing the cells and the organelle and everything. And there are so many variations, like you can use confocal microscopy, you can use high content image analysis, and all those things. The another platform is the flow cytometry. the antibodies, either with the primary antibody directly or with the secondary antibody. So this is one of the very common principles in FACS. This is just there are so many other fluorochromes directly also being used, but most of the cell-based applications, these kind of antibodies are used. And ultimately fluorochromes. So fluorochromes are basically the key components in flow cytometry. And now if you could understand this principle of antigen, antibody conjugated with a fluorochrome or some coloring molecules, this is a common principle in Western blot, immunofluorescence analysis, in most of the confocal analysis, and protein microarrays. There is a technique called immunoprecipitation. You can like pull down the protein complexes. There also the principle is the same. There is another technique to pull down the protein DNA interactions. That is chromatin immunoprecipitation. There also the principle is same. Primary antibody, secondary antibody conjugated with something. In some cases it's fluorochromes. In some cases it could be something else. So the basic logic is you have an antibody conjugated with a fluorochrome, which is basically used to tag the proteins or to tag the cells. And another one, in flow cytometry, I think Mukund will be talking much about this one. In flow cytometry, you would be analyzing the molecules. Okay, so it's basically also a separation technique. Like we have two types of molecules with the different, the granularity and everything, that also can be analyzed. You have a lot of molecules in cell lysates or in a group of cells. You have a lot of molecules. You have a lot of cells. But in flow cytometry, you are basically interested in analyzing a particular type of cells or particular protein alone of your interest. So that is basically being done by tagging the protein with some antibody. And if it is some other molecules, it's like, cells, it's based on the granular, it's like, maybe based on the cell size, and the other things also you can separate. So fluorochromes are the key components in the flow cytometry. And you all know about, the excitation and the emission wavelengths of all the molecules. And mainly in flow cytometry, not only in flow cytometry, again, the logic is the same in confocal, fluorescent microscopy, and in many other platforms. And you can use varieties of fluorochromes. If you want to tag a protein with the red fluorochrome, you can do that. If you want to tag with the green fluorochrome, you can do that. You have like n number of molecules which are basically fluorochromes. I think again for students, as I said earlier, understanding the principle is a very, very important aspect. Everybody cannot come and solve all the application things. I think we have to understand the principles. For example, here you can take, I think, if a chemist student or a biology student in collaboration with a chemistry student can make a new fluorochrome. If you make a new fluorochrome, you can patent it, you can market it. There are a number of possibilities. So you can see the slides. There are at least some 40, 50 fluorochromes are there. Across the entire spectrum of wavelengths, you have different fluorochromes with different features, different excitations, different emissions. And you know about the Cy3, Cy5, propydium iodide, and there are a lot of things actually, phycourethane. So many fluorochromes are there. And all these are basically, depending upon your experiment, depending upon your combinations of the molecules you are analyzing, you can basically select the fluorochromes. Okay, again, just a recap, fluorochromes are very important. And in many of the applications, cell-based applications, antibodies also important. You are tagging the cells with one particular antibody, and then subsequently, you are basically marking with a fluorochrome. So one of the basic applications in cell biology or in mammalian cell biology or in cell culture-based experiment is understanding the cell cycle phases. For example, in drug discovery, people will be using a lot of drugs. So you want to understand that what is the best drug and what is the molecular mechanism of a drug on a cell. So what you can do is in a dish, you can grow the cells and then add a drug of your interest, and then you can try to see that what is the impact on cell cycle phase distribution. If you can, very well the basic cell cycle phases, G1, S, G2, and M phase. And depending upon the cell cycle phase, the DNA content of the cell would be varying. Here you can use the dyes like propydium iodide or something else, and those would be basically would be showing the as you can see in this graph, the DNA content is basically measured in the flow cytometry through the fluorescent intensity, and based on the DNA content and based on the whether it is it is a 2N or it is a haploid genome, and those kind of things can be you can identify in the facts. So DNA content can be identified, and the cell cycle. So imagine you have in a tube you have 1 million cells. Then you can understand that out of 1 million cell, how many cells are there in G1 phase, how many cells are there in S phase, how many cells are there in G2 phase, you can understand. So by using facts, you can understand the distribution of cell cycle phases. And another application is ploidy, mainly in some plants and also in some fishes. The ploidy is a big issue. And in humans also, in some diseases and disorders, you would see some aneuploidy kind of issues. And by using DAPP like kind of fluorochrome or dye, you can stain the cells. But another important aspect in cells is the permeability. One is now we have seen that there are large number of fluorochromes are available with a different excitation and emission. And you choose the fluorochrome of your choice. Similarly, you can also use different dyes, and you can measure the content of the DNA. And in this case, like for example, you can see here, diploid cells are here and tetraploidy cells are here. And in this case, even some 8 sets of chromosomes and then 16 sets of chromosomes are there in some extreme cases, and that can be analyzed by facts. And there are some studies. Even people have shown the aneuploidy. Just you have one extra chromosome or loss of one chromosome, that because that would have impact on the DNA content of the cells, and that can be analyzed by using facts. So this is a platform not only for the basic biological analysis and also for the clinical investigations and diagnostic investigations. So another method which is being done often in the laboratories is live dead cell analysis or analysis of the apoptosis. So normally what happens in apoptosis, but again, why I am talking about this one is, as science students, we should always, while we do classes in MSc, in the first semester in molecular biology, we teach like, if you are using one particular solution, you should know the reason for each and every chemical. For example, if in a lysis paper, if you are using sodium chloride, you should know why you are using sodium chloride. Because there should be a reason, there should be a mechanism for that. And similarly, EDTA, why are you using? SDS, why are you using? Similarly, and here also, like as a students, I always encourage you to understand the principles, because that would give a lot of joy to you, and then subsequently give enough room to modify your experiments, plan your experiments, and in future, when you become an independent researcher, that would create an opportunity for you to design the experiments and even create the reagents and even to develop the methods. Okay, so you guys, I suggest always, whenever you talk about a technique, you try to understand the principles. But for example, in this case, the principle of apoptotic cell analysis is, while the cells are undergoing apoptosis, the phospholipid which is normally present in the inside membrane is like getting evaporated, and so that phospholipid comes out of the membrane. So there is a dye called annexin 5. There is also peptide actually. So that peptide annexin 5 can bind with this kind of phosphatidyl serines, which is like coming out of the membrane, and that is subsequently tagged with another fluorochrome. In this case, phycoerythrin. So phycoerythrin is a fluorochrome. Then by using appropriate excitation and emission, you can visualize the cells which are. So imagine you have 1 million cells. Out of 1 million cells, in how many cells this kind of phosphatidyl serine is outside, and so you can label with annexin 5 and then fluorochrome. You can try to see that. So in a mixture of 1 million cells, how many cells are being. Now you can use a separate another dye to track the live cells. For example, in this case, acetaminophen 7-AAD is used as a vital dye. So by using two different dyes with the different fluorochrome properties, you can track, understand, how many cells are live due to the treatment of a drug. So in different time points of treatment of a drug, how many cells are live, how many cells are dead, how many cells are undergoing for apoptosis. So you can track the live cells, early apoptotic cells, and dead cells and everything. So in drug discovery research, these kind of experiments would be very, very much useful. You can understand the apoptosis, you can understand the live cells, you can monitor the cell proliferation. There are n number of methods. I'm not going to talk about all the things. Yeah, this is the same thing, live cell, dead cell analysis. And another important aspect is you can analyze the protein expression. You all know that about the Western blot. For Western blot, what you have to do is you have to isolate the protein, you have to run in a gel, and then you have to do a Western blot, transfer to a membrane, and then detect it. But here you can take the cells, and if they are homogeneous cells, and that directly you can look for your protein of your interest. Again, one more thing is you have to always think about the permeabilization. Normally cell membrane is not permeable, and you have to fix them with appropriate fixing agents. And if you want to study the intracellular proteins, you have to use such kind of agents. And so there would be a permeability in the membrane, and your dye and antibody can enter inside. And if you are analyzing the plasma membrane, then you have to use different fixatives. Those are the other kind of principles which you need to think of it while you want to learn flow cytometry. So in this case, normally what people do is, they always, in the experiments, they will be using green fluorescent protein or some other protein. Normally, if you want to express a protein in a cell, what people do is they will take the appropriate plasmid construct with appropriate promoter, open reading frame, and everything. And most of the cases, along with the protein of your interest, people also would have the GFE marker. Now, if you are overexpressing one particular gene in a cell, and you are expecting your protein interest, but the GFP you will be using as a surrogate marker. If GFP is expressing, which means that your protein is also expressing. So now in this case, what you can do is, like if it is a GFP, the excitation and emission based on the as you see here, you can try to monitor. Whenever you do the transfection in the cells, out of million cells, probably maybe few thousand cells also alone might have been taken your plasmid. So all the 100% of the cells would have your protein of your interest, only maybe 10% or like 5% of the cells. Now in flow cytometry, you can analyze only, that is called sorting. That is not sorting, that is gating. You can just gate only that particular percentage of the cells, and which are they have taken the plasmid, which are expressing your protein of your interest, which you are understanding through the expression of GFP. So as you have seen here, so when you have millions of cells in your test tube, you can analyze the fraction of cells which are basically expressing your protein of your interest. So protein expression, so normally this experiment would, to my knowledge, it would take probably 1 or 2 hours. But if you want to do Western blot, conventional Western blot, it will take 2 days. Okay. So again, these kind of things can be done also in the clinical settings. You can take the blood, maybe 1 ml blood, and from that you can analyze a particular protein expression. So protein expression can be done to some extent with flow cytometry. And you can also understand the subcellular location, because we all know, always think about. But most often another important aspect is whether the protein is in the cytoplasm or in the nucleus or in the membrane, or it is secreted out. And by using appropriate controls and combinations of antibodies and dyes, you can also understand the subcellular location of the proteins. So subcellular location of the proteins, at least whether it is a nuclear, whether it is cytoplasmic, or whether it is membrane, that is can be done with subcellular localizations. I think this can be done very well with confocal and fluorescent microscopy. But to some extent, whether it is intracellular or extracellular, that can be done with flow cytometry. And another one is the protein-protein interaction. Protein-protein interaction is, in biological system, most of the times the proteins are not acting alone. Some 2 proteins or multiple proteins join together and they interact, and then they do the biological function. So in this case, if 2 proteins are interacting, and there is a technique called as FRET, fluorescence resonance energy transfer. If 2 molecules are very close, I think imagine one molecule is having the excitation of 500 and the emission of 550. Now the second molecule, second fluorochrome, would be having the excitation of 550 and the emission of 650. So you would be exciting at 550 in your machine, 500, but you would be getting the emission at maybe 600 or so. So that is that's a basic principle in fluorescence resonance energy transfer. And in flow cytometry also, within the cells, you can try to study the protein-protein interaction based on the FRET to some extent. Again, these kind of things can be done well in some other platforms, but in flow cytometry, the advantage is you can done in the blood, you can done in the cells. That is advantage, cells in suspension. Okay, and another very recent aspect of flow cytometry is because so far many of you might have isolated RNA, DNA, protein, and you will be taking mg of the 1 mg or 10 mg of a tissue, and then you will be grinding them and then getting the whole protein. But in 1 mg of tissue, you would have 1,000 types of cells. You have mesenchymal cells, you have fibroblasts, you have immune cells, and in immune cells itself, you will have so many types of immune cells, T helper cells and CD4+ cells, whatever, blah, blah, all the cells you would have. So biology, so far people were like, analyzing as a mixture of tissues, but now there is a possibility, there is a new area in genomics. People say single cell genomics, rather than like, analysis taking 1 mg of tissue, which is highly heterogeneous and it has a mixture of cells. And instead of that, what people are saying is now you disintegrate them, make them into single cell suspension. And in a flow cytometry, so far I have not mentioned anything about sorting. In flow cytometry, you can sort the cells. Because if you once you tag a cell with a fluorochrome, then in a 96 cell plate, you can sort like 1 cell per well. You are taking a 1 mg tissue, you disintegrate with appropriate proteases and whatever, and then subsequently you can make them in a suspension of cells. And if you want to separate out only CD4 plus cells, there is a protein which is expressed only in CD4 plus cells. And then by using that antibody and then one fluorochrome tagged with that, you can, among millions of cells, you can tag only, fluorescently tag only CD4 plus cells. Now, the CD4 plus cells alone can be separated out in FACS by FACS sorting. So normally what people do is in a 96 cell format, in each well, they will put 1 cell. Subsequently, you can take the cells and you can analyze. So by this way, now heterogeneity can be delineated from in anything. It is applicable for like, protein studies, DNA studies, metabolome studies or biochemical pathway studies, rather than studying the whole tissue. For example, in the case, this is applicable in cancer also. Because so far, like, people will always take a tissue, heterogeneous mass of tissue, which has so many types of tissues and so many types of cells. You can separate out them by FACS and then do that. Okay, so this area is single cell genomics. That's a big area. And now what people are doing is they are like, doing RNA-seq and then trying to separate the cells. What are the cell types? For example, any experimental sample, you take it. And then if you analyze, you will have heterogeneous types of cells. Now, by using FACS, you can sort out the cells. And then, each of the cells you can basically investigate separately. You can do RNA-seq separately in each of the cell types, or you can do any protein studies separately in each of the cell types. So you will understand the constituents of a complex tissue. So that is being possible. In the last 2 years, a lot of papers are coming in that line. You can see here. This probably from blood, but again within blood you will have different types of cells, like monocytes and myeloid cells and CD4, whatever. By using different combinations of, so in fact you can always use a combination of 10 fluorochromes. That is called multiple colors. Mukund will be talking much about that. You can do multiplexing. You can label 5 proteins at a time, 10 proteins at a time, and then you can separate everything. Okay. Yeah. So this is like take the cells, you run a FACS. And you basically analyze the thing. And as I said, in 96 cell plate, in each well you can put 1 cell and you can expand the cells basically. And subsequently now you have like, from the tissue, you have taken 96 cells individually. You can expand them. And then subsequently, if you want to do PCR, you can do PCR. And if you want to do protein expression analysis, you can do analyze. And you can study the heterogeneity in a better way. Okay. And another one thing is genome-wide screens. Nowadays, what people are doing is now we have the whole human genome sequence and whole human genome sequence annotated. The next area is like to understanding the functionality. So what people are doing is the genome-wide screening. You can screen like, you can take 20,000 lentiviral particles, each one having the short hairpin RNA for one particular gene. And you can infect hepatitis with the human cells, and then you can like FACS sort the cells which have taken up the lentivirus, and then you can analyze them. Or if you are interested in, you can design n number of experiments in that. That depends on your phenotype. You can study for cell proliferation, you can study for apoptosis. So large number of genome-wide screenings can be done with the FACS. Similarly, genome-wide mutagenesis. So if you want to make a mutant, you take the cells and then you radiate or you use some chemical mutagens, and then subsequently you can look for some phenotype. If you are looking for a phenotype, like the cells which are going faster, or the cells which are expressing another protein, or the cells which are resistant to a drug, you can always select a phenotype by a small experiment, a small short-term experiment, and then by using FACS, you can just sort out the cells. You can pick up your selections. Normally, this kind of mutagenesis and the mutant selection is like it's a year project or several months project. But with this kind of FACS sorting, in a pool of cells, you can select your interest of mutants of your interest, and you can go ahead. I think I'll come to the last part of the thing, And nowadays, like, everybody is talking about CRISPR-Cas9, and with the CRISPR-Cas9 you can do knockout. Knockout means removing a particular region of the gene or the genome, or like you can knock in. If you want to introduce a mutation, you can introduce. If you want to introduce a foreign gene, you can introduce. Typically, CRISPR-Cas9 vector would be like this. It would be coding for guide RNA, which you have to design. For example, if you want to delete a gene of your interest, then you have to design the sgRNA for the gene of your interest. And you need a Cas9 protein. I don't want to go into the CRISPR-Cas9 logic. You need to have sgRNA, and then a Cas9 protein is necessary for doing this editing. So you need to have the Cas9 protein, and also you clone. You have in the downstream GFP protein. Now you take if you want to delete a gene, or if you want to introduce a point mutation in your cell line, or if you want to knock in something, so you design sgRNA. There are different vectors available depending upon the purpose of your interest, and you can clone them in the vector, and then you transfect the cells. You take human cells and transfect. Now, as I said, whenever you do the transfection in a million cell, only few thousand cells would have taken the plasmid. Remaining probably several lakhs of cells are without plasmid. But there is no way to analyze all the cells, because you want to pick up a single cell which has taken your plasmid. Not only that, it has, because these kind of knockouts are happening in a it may not happen in a perfectly. If you screen 100 clones, one of the 100 clones would be having the knockout of your desire. So you have to screen 100 cells, but what I'm telling is, out of the transfected cells, you have to screen 100 cells. But now, if you take the whole set of cells in the Petri dish, you will have millions of cells. So at least you can select out only the cells. For example, as you shown here, you transfect, and then you take the cells which are green, because they have taken the GFP, which means they have also taken the plasmid, your sgRNA construct. Now, then you seed them into 96 cell plate in each cell in one well, and then you can expand them and you can do your experiments. So by doing your phenotypic screen, or by doing your molecular screening, you'll be able to derive a cell which is of your interest. You have knocked out the region of your interest, or you knocked in something, or you have created a point mutation. Okay. So now I think probably this is the entire lecture. You can study n number of cellular phenotypes and cellular biology, cell biology, and protein expression, protein understanding. You can also create knockouts, because this experiment normally with the help of facts, you can do this probably within a week. Within a week, you will have your knockout in hand. Actually, if you go to nowadays many companies are selling knockouts also. One knockout may be costing 2 lakhs or something like that, but which can be also only if you have a flow cytometry, you can make it in your place, and you can do a lot of biological experiments. So as I said earlier, people are doing a lot of genome-wide functional screens also with facts. So I think probably I'm done with this. So thank you very much. So thank you very much for your time, and I'll be glad to answer any questions. கேட்க இந்த கேட்க சொல்ல வேண்டியதான் சார், அவ்வளோதான். Yeah, any questions please? If you have any questions, please ask, or else you message it. I will answer to all the questions. So hope the lecture might have been useful to you. So I gave an overview of some methods. Okay. So if you have any questions, please send, please text it. And yeah, in the chat you text it, or you send a mail to me. Definitely within this evening, I will give you the answer for your question. Okay. So if you have any questions, you always ask. Sir Mukund sir, can you please take over the session?
Sure, sir. Thank you so much. I think that was a wonderful session. You covered a lot of applications. Perhaps we could have discussed on flow. Also, helped me a lot in my own presentations. But I'll stick to a lot of principal aspects of how you look at a flow cytometer instead of just looking at it as a black box. So let me present my screen. Right, is the screen visible, sir?
Yeah, yes, sir, fine.
Right. So at the outset, thank you so much for this opportunity. I really appreciate, because this is one of the best ways we can, spend our time productively during this crisis time. So the outline of my lecture today, and in fact tomorrow as well, would be introduced to you basic aspects of flow cytometry in the first place, and how different components of flow cytometer come together and work for you. And then a little bit of, do's and don'ts on experimental biology as well. Tomorrow the session will be entirely on how to actually do, let's say, a 10 color study, or for instance 20 color study, when you're looking at 20 different antigens all at once. So how do you design such experiments? How do you plan such panel design constructs? So that's the theme of tomorrow's session. All right, so this is a standard slide that we put across because we work in a commercial setup, and therefore this slide deck might include some bit of, intellectual property or confidential information, sharing of which would be prohibited without prior permission. Okay. However, if you need any information from the slide deck, you can always write to me. I'll be happy to help you on this. The outline for the next 50 minutes, for the first session of my talk today, would be to talk about the flow cytometry itself as a principle, and then the components, and then how do they all come together and work. And then we will discuss in brief about immunophenotyping. Remember, whatever Professor Kumaresan talked about right now in terms of single cell genomics, or in terms of even cell cycle, I think everything boils down to this very important fundamental technique of immunophenotyping. Of course, there are other methods for immunophenotyping. We will focus on flow cytometry based immunophenotyping. And like also I have included a guideline for presentation. These days when we annotate or when we go for publications, there are guidelines around how to do that in terms of flow cytometry experiments. So I will introduce to you that as well, and then share you the article as we move along. Now, the first question before we head to flow cytometry is to answer what really is the difference between, let's say, an image cytometry and flow cytometry. Because cytometry in essence is measuring cells, or measuring the properties of cells. What properties, we will look at it a little later. But then when you talk about image cytometry, you're basically trying to image the cell, or it is a very static experience, okay? Because you need cells not floating, but then adherent, sticking to some slide, and then you sort of image them. But the good thing is you can also look at the morphology, you can look at the counts, for instance, and then you can also look at perhaps the localization of antigen, which is the most important aspect of imaging. Can we do this localization of antigen on a flow cytometer? The answer is no right now, although there are a couple of papers which talk about localization in an indirect way, but still microscopy or imaging I would say is the gold standard for localization of antigens. Now, if I can't do localization of antigens, and if I can't look at morphology, why do we do flow cytometry then? Are there any advantages to flow cytometry? Let's take a look at that. Before we go there, I will not try to give you the textbook standard definition of flow cytometry, because that does not help you understand the conceptual learning. What is really important is we are trying to measure the properties of cells as they are in motion. Now, the key question is what properties of cells, okay, or what properties of particles? Two predominant properties would be physical and chemical. And when I say physical, I mean size and shape, or how complex a particular particle or cell type is from within. Okay, I repeat, physical properties, size and shape, and how complex a particular cell type is from within. Okay, and then when it comes to chemical properties, you can look at protein expression on the surface of the cell or within the cell if they are cytoplasmic, or perhaps you can also take a look at the nucleic acids themselves, DNA and RNA both. Okay, now these are some of the properties that you can measure using a flow cytometer. So I said it's a technology that measures properties of cells. The next question is, as they are in motion, okay, we will look upon how do we make the cell move in motion in the next couple of slides. This is the key definition of flow. But then they highlight the importance that we have to understand in terms of why flow cytometry is its rapidity. I can analyze as many as 100,000 per second. I repeat, 100,000 per second when it comes to flow cytometry, as opposed to, let's say, even if I give you 500 cells to image, you will take all day in front of a microscope. Okay, microscope has its own advantages, and so does flow cytometry. The most important advantage of flow would be the speed at which we analyze the cells. Okay, so this is the classic definition of flow: measure the properties of cells as they are in motion. Now, the next question is, what are some of the ranges of particles that I can analyze on flow? Can I look at, let's say, an atom? The answer is no. Can I look at a protein? Yes, we do look at a protein, but then in conjugation with some other fluorochrome. Today's time, there are instruments, sophisticated instruments available which can look at, let's say, a nanoparticle at 30 nanometer or a nanoparticle at 100 and 150 nanometer. Something in biology we call as microvesicles or extracellular vesicles or exosomes at 150 nanometer also can be looked at using the flow cytometer, as opposed to the standard blood cells or standard plant biology that we have been doing all these years. So if I were to give you a size range, it would be in today's time, let's say from roughly from 100 nanometer to all the way to your protoplast biology. So that's what flow cytometer has come to in today's time. Okay, so this is an idea about range of particles one can use using flow cytometry. Anything bigger than perhaps let's say about 100 micron is really not feasible for flow cytometry. Now, I talked about what do we look at in terms of the properties of the cell. Okay, I mean, I can look at the cell surface and look at the expression of certain CD molecules, as we say, which will sort of help you phenotype different cell types in the body, particularly immune cells. You can also look at when you induce, or when you, let's say, add a drug, what does it do downstream of the cell? What does it do to the cell? What does it do to your, let's say, replication or your cell cycle? So I can really study the cell health or cell processes in terms of cycling, in terms of apoptosis, or the proliferative capacity of, let's say, a drug to induce proliferation in the cells. All these can be looked at when I try to analyze a cell by flow cytometry. I mean, this is just an indicative list. You can do many things like Kumar sir pointed out in his session. You can also look at the fluorescent proteins if you're doing gene expression studies, for instance, or if you're looking to study transfection biology. You can still employ a lot of these GFP, RFP, YFP proteins available for flow and do your biology. Okay, so this is just a gist of what one can analyze of a cell using flow cytometry, and these are both chemical and physical. Physical, when I say, I mostly talk about the topography, the size and shape of the cell type. Okay, the important point to note here is, I mean, some bit of my session will sound like a bit of physics, but it will be elementary in nature, so don't worry. But it is very important that we understand this if we are to understand and appreciate facts a little bit better. Okay, why do we have to know electromagnetic spectrum? Two reasons. Now, when you're trying to set up your experiments using fluorescent reagents, remember every fluorescent reagent will have a spectrum. When I say spectrum, I mean excitation wavelength and I mean emission wavelength. Okay, so every reagent you buy will have an excitation wavelength, and it will need a different type of laser for excitation. And every reagent will also come with an emission wavelength, and that will tell you what detector should I be looking at in my machine in the lab. So this has practical utility. Okay, roughly what we operate or where we operate is mostly in the visible range, although we have lasers in the IR range today. I mean, it's from our company as well, but then very limited applications as of now. But for most biological applications, what we have had is in the visible range, also UV 355 and 375 nanometer range. So that's the reason why you have to understand this slide well to know for a fact that every reagent will come with a spectrum, and it's important to know the spectrum to know whether those reagents can be utilized on my flow cytometer in my lab. Okay, so this has practical utility. The other important aspect is also fluorescence. I mean, a molecule absorbs laser light or any light, for instance, and it will go to higher state because it is now full of energy, but it can't stay there because it's not stable. It has to come back to the ground state. Now, while it is coming back to the ground state, it will lose some energy in the form of heat. I repeat, it will lose energy in the form of heat. Now, the fact that it is losing energy, which means energy and wavelength are in inverse ratio. If energy comes down, the wavelength goes up. Okay, so what was here at the beginning, let's say about 400 nanometer, would be now 450 nanometer. So there is a change in wavelength. This is what we call as fluorescence. Okay, you have an excitation maxima, you have an emission maxima, And if you shine a light, let's say on a molecule, it goes to higher state, comes back, emits in the longer wavelength region. Okay, so there is a change in wavelength. Now that's fluorescence. This is the standard Jablonski diagram. What are the components of a flow cytometer? This is in simple way, because I saw that there are a lot of students also on this webinar, so I've tried to simplify this. In the next slide, I'll try to connect you to what exactly a flow cytometer looks like. So every flow cytometer will obviously have a light source, okay, and predominantly the light source will be lasers. Why lasers? We'll talk about that a little later. And then you should have a flow cell. Now this is where a cell and a laser light meet. I mean, this is the meeting point. They talk to each other only in this chamber called as flow cell. Now once the cell and the laser light meet, obviously they will give out some signals, whether it is chemical or physical, okay. Now, because we are not doing flow just to look at one fluorescence, obviously there will be multiple wavelengths. You need to have different filters to filter different wavelengths. So the signals are here, the wavelengths are being filtered by the optical system, and then they are sent to detectors. Once they reach the detectors and perhaps let's say electronics, it is still analog signal. It is 0, 1 machine language. You and I cannot understand this language. Therefore, the electronics box on a flow cytometer will convert this language, digitize this value, and give it to us in the form of a human readable format. Okay. And finally, the display. I repeat, you have a light source, lasers mostly, and then you have a flow cell. This is where the cell and the laser light meet. And then they all go to optical system, get separated based on the different filters we use, go to a photo detector, and then they have to go to electronics for digitization, and then fed into the computer. So this is the scheme of things that happen on a flow cytometer. Now, how does it look exactly? I mean, when I say flow cytometer in simple lay terms, for instance, I talked about light source as lasers, where I'm pointing out right now, and this is a flow cell, okay, where you inject your sample from the top, and then the laser light will hit the cell. They talk to each other, signals are out. Before the signals can reach all the detectors here, they have to be filtered using various filters. I'll talk about that a little later. So once the filters are filtering different wavelengths, they go to different detectors, but it is still analog signal 0 and 1. You and I don't understand, so therefore they have to go to electronic box. Finally, the output will look like that. Okay, so this is the scheme of things more scientifically rather than more in layman terms. Now, we talked about properties of cells being measured as it is in motion. Okay, we still haven't answered how do we make them in motion. We'll come to that. Let's figure out what properties in the first place. We'll start with physical properties. Okay, can I measure size on a flow cytometer? The answer is yes. Is it absolute size? The answer is no. When I say absolute, I cannot say it is a 10 micron cell or a 20 micron cell. Okay, no, you can't do that on flow. It's not a sizing machine. It is relatively, in terms of relativity. When I say something is small, I need to have something as big. So there has to be always a comparison. Okay, that's why I say relative size. Now this is how the cells are flowing top down. You have a laser which is hitting it on the left side. This is just the design aspect. Laser hits the cell, and in the direction of the light, you have something called forward scatter. I repeat, in the direction of the laser light, 180 degrees straight line, you have forward scatter. Now this tells you about the size, the approximate size of a particle or the cell that you're measuring. Okay, anything in the direction of the laser light will give you forward scatter. Right. Now, can the scatter change depending on the size of the cell? The answer is a yes. I mean, if you have a big cell, obviously there will be proportionately bigger scatter. And then if you have a small cell, like I have pointed out here, you will have a smaller deflection around the cell. Remember, this is a cone of light. The laser light is not straight away hitting the cell. Actually, it is the lenses that give you the shape, the beam shaping, and which will eventually hit the cell, and then you will see the signal on the detector. So it's the cone of the light, usually plus minus 8 degree from the central axis. I repeat, laser light hits the cell. In the direction of the laser light is what you call as FSC, or forward scatter, or forward angle light scattering also called. This tells you about the size of the particle being measured. Big cell, bigger scatter. Small cell will have a smaller scatter. Okay, the example, if I have to give you in terms of everyday example, is if you have a bigger circle, if you're traveling, let's say in the city of Madurai, and then if you have a bigger circle, obviously you'll have to take a bigger round using your car. Perhaps you have to take a bigger Why not any other degree? Is there anything divine about 90 degree? Remember, this is just an engineering aspect of the flow cytometer. Why do I say that? For instance, if you take the sky appears to be blue, whether you're in India or in US or any part of the world, it is blue everywhere. Why is that? Because light scatter is in all directions. It's 4 pi r. It's the sphere of the light. So if the light scatter happens in all directions, I can't put a filter everywhere and then collect light. So this will limit my design aspects. Okay, so considering the design limitations, we collect by convention in 90 degree. There is no hard and fast rule that you have to do it at 90. Okay, all said and done, most cytometers, even in today's time, collect side scatter at 90 degree, and this is also where you will collect fluorescence. Now, you looked at FSC, you looked at SSC. Can I utilize both of these things? Size on one axis, granularity on the other axis, and then let's say distinguish my cell types. If I give you blood sample, chop the RBCs, and I ask you to do, let's say, a distinction of lymphocytes, monocytes, and granulocytes, can I do that just using the scatter properties without adding any reagents? The answer is a yes, because every cell type will have a size, and every cell type will be complexity will also change basis the cell type, right? So on the x-axis you have forward scatter, which talks about size. How do I read this plot? The intensity will increase from left to right. I repeat, the intensity will increase from left to right, which means the sizing will increase from left and as we move towards right and right. Okay, same way for side scatter, the intensity will increase from bottom up, which means the complexity will increase from bottom all the way 0 to all the way to 100 up. The more complex the cell type, the more higher it will be on the plot. Okay, now let's figure out, if I give you human blood, okay, K2EDTA collected human blood, I chop off the RBCs, and then I ask you to identify different populations just based on the scatter profile. How would an RBC look like? RBC, as we all know, is roughly about 5 micron, roughly. Okay, now that's the area where RBC will populate. Okay, now if there is anything bigger than RBCs, it has to be moving to the right, correct? Now what's bigger than RBCs in your blood sample? Perhaps your lymphocytes, monos, and granules. Let's see, where are the lymphocytes? These are your lymphocytes. Why? Because they are bigger, they are on the right angle to the RBCs here. They're also slightly complex, if you look at the y-axis, than RBCs. Now let's take a look at monocytes. I mean, they are even bigger than lymphocytes, roughly about 12 micron, and most obviously they are more complex than lymphocytes. Therefore, they are moving up on the graph as well. They are moving right and they are moving up. Okay, and the biggest of them all and the most complex cell type is perhaps granulocytes, although they don't differ much in terms of size with respect to monocytes, roughly the same size, 1 micron here and there. But granularity is pretty high when it comes to these granulocytes and mostly neutrophils. You can see they're all the way up on the side scatter. Okay, now this is how simply you use a forward versus side scatter to identify different cell types in your WBC populations. How do I read, for instance, a fluorescence plot? Let's say now I add a fluorescent reagent and I want to do some bit of studies on lymphocytes, subset analysis. I want to look at T lymphocytes, I want to look at B lymphocytes, and etc. How do I read the plot in the first place? Again, the same logic. Any fluorescent plot on a flow cytometer has to be read this way. The intensity will again increase from left to right. That's what the arrow indicates. Okay, so if you have very few antigens and very few antigen antibody bound to a cell, obviously the intensity will be low and it will be on to the left of your axis. Okay, and if you keep adding more and more reagents and if there are more and more antigens available for binding, definitely the intensity will increase from left to right, from medium to all the way to bright. So again, the logic of left to right either on scatter or on fluorescence will hold good. Okay, now if you remember, we talked about properties of cells, we talked about size and shape, we talked about how to read a scatter plot, how to read, let's say, a fluorescence intensity plot. We didn't answer still one question as to how do we make the cells move in motion. Okay, we said measure properties of the cell, that's fine. How do we make them move in motion? So every flow cytometer will have a component called as fluidics. Now this is where you will have your sample tube, you will have something called sheath tank. What is sheath? Essentially phosphate buffer saline pH 7.2 with some anti-growth additives to prevent bacterial and fungal contamination in your machine or in your sample tubings. Okay, PBS pH 7.2, mostly free of calcium and magnesium because it helps clumping. Now, every flow cytometer will have a sheet tank. Basically, let's say either a plastic container or a steel container, let's say on a sorter. So you will have sheet fluid inside this tank, which is essentially PBS, and then you have a sample tube here, let's say a 5 ml tube with about 1 ml sample in it. Now both of them are sitting still. How do I make them move in motion? You apply air on top of it, which means compressed air. Okay, this is like pumping your cycle tires. Once you start blowing air by capillary action, the liquid starts traveling through this sample tubing and it will go all the way to flow cell. I repeat, you pump air in. Because of the pressure, by capillary action, now the fluid starts traveling and it will reach flow cell. Similarly, for your sheet tank, because the tank is now compressed, okay, this is pressurized, so to say, the liquid starts traveling even from here and it will go to flow cell. I told you flow cell is a point where your cell and the laser light meets. So now the cell is waiting there. If you hit it with the laser, the interaction will happen. The other key aspect that we have to understand here is, for instance, if you're analyzing blood cells, there are millions of cells, correct? I mean, you will have a lot of cells flowing through. So initially, when you inject a sample, you will have many cells. But if you notice, after a while, they are all lining up one by one. Okay, which means they're traveling one by one, or they're showing themselves to the laser beam one by one. Okay, this is called a single file, or this is called a laminar flow. The process of making cell move one by one in front of the laser beam. Okay, this is called hydrodynamic focusing on a flow cytometer. This is the gold standard principle of flow cytometry, and this is exactly the heart of flow cytometry, so to say, because if you're not getting single cell file, then you need not do flow cytometry at all. So the whole point of doing flow cytometry is to be able to look at a cell on a single level. Otherwise, ELISA can also give you pretty much the same information, or perhaps Western blot too, because it will give you a whole lysate, the aggregate information. Here you look at cell by cell. The picture on the right is the actual flow cell. This is how a flow cell will look like. This is from a machine called Cytoflex that we have. Notice the conical shape. Remember, but this is not physical separation. This is a liquid interacting with another liquid, a sheet fluid interacting with sample. Okay. Now I talked about pressure. Remember, I said you have to apply air pressure here and you have to apply air pressure on the sample tube, and then they all go and meet in the flow cell. Question is, do they not mix with each other? Obviously, because you're running sheet fluid, which is PBS, and you're running your sample, which is already diluted in PBS or some other buffer, they all go together and sit in the flow cell. Do they mix? If they can mix, of course, but if the pressure differential is not maintained. Now, what do I mean by pressure differential? In flow cytometry, the sample pressure, I repeat, the sample pressure is always higher than sheet pressure. Okay, sheet pressure on most analyzers. When I say analyzers, machines which can do analysis but cannot sort. On most analyzers, sample pressure is always higher than sheet pressure. Okay. Now, if I talk about in terms of pressure, I mean, some of you might get confused because these are engineering terms basically. How do I, as a user, when I sit in front of my flow cytometer, change these fleshers? Because if I have sample pressures, I give you 3 different flow rates. Every manufacturer rather gives you 3 different flow rates to control the sample volume that you push to the flow cell. One is low, the other is medium, the third one is high. What happens at low is you're pushing roughly at about 10 microliter per minute. I repeat, 10 microliter per minute of sample is being pushed from the sample tube to the flow cell. Okay. Now what happens when you switch to medium? You will push more volume, 30 microliters, so on and so forth for high at 60 microliters. The question here is, if you operate on low pressure, your sample stream, the other adjacent streams in blue here are sheet fluid, the central sample stream, it looks like the cells are traveling one after the other almost. Okay, more than 99% of the cells are traveling one by one, so therefore single file is maintained. Therefore laser can look at the cells, you know, in a very relaxed manner. Now the problem with high pressure is a lot of cells are flowing all at the same time, which means you are disturbing the laminar flow right now. Then if the essence of doing flow cytometry is to look at a cell one by one, why do I, as a manufacturer or any manufacturer, give you different flow rates? Okay, because it is misleading then. The answer to this question is, Sometimes when you do biological experiments, you will not always not have the same cellular concentration. Sometimes when you, let's say, have pediatric samples, if you're doing pediatric immunology, you will not always have the same amount of sample available because these are clinical samples. So when you have very little sample and then you want to be done with analysis, not spend the entire day doing just one analysis, that's when you switch to high pressure and then quickly be done with your acquisition. Okay, so this is to do with your biology. But then if you operate on high pressure, sometimes your CVs will go for a toss. We completed fluidics now. We now understood how do we make the cells move in motion. Remember, this is just one way of making the cell move in motion. There are instruments like our own Cytoflex which will operate on peristaltic mechanism. Okay, at one end you will suck the sample, at the other end you will suck the sheath fluid and then make them meet together in the flow cell. This is by peristaltic action. Okay, and then there are syringe based fluidics machines as well. So this is the most common and the most, in fact, traditional way of doing things, applying pressure and making the fluids move. Now let's go to the optics. The optics, I said, I mean, there are two aspects. One is the filters which collect the different wavelengths. The other is the light source itself, the input. Okay, like I pointed out, the light source are always lasers. In today's most flow cytometers, of course, they started off with mercury arc lamp and other source of light back then in 60s and 70s, but today you have mostly lasers. Now, why lasers? Because lasers give you a coherent source of light. They're all in the single phase. Okay, when I say 488 nanometer, I usually get 488 nanometer. Okay, and I don't get any other wavelength. Because of this purity or because of this coherence is why we use lasers on flow cytometry. Okay, and what are some of the different types of lasers people use in flow cytometry? Like I told you, the entire visible range, mostly Vibgyor range, violet, indigo, blue, yellow, green, red, and today's time we have also an IR laser, and we also use UV lasers for almost all our biological applications. When I say UV, violet, blue, what I mean is the wavelength itself. Okay, when I say UV, I mean 355 or 375 nanometer. When I say violet, I mean 405 nanometer. Remember, whenever you talk about lasers, never skip talking about the nanometer because that's the measurement, that's the unit. It says no longer presenting. Is the screen visible, sir? Hello?
Yes, sir. Yeah, it's visible. Sir.
Okay, I got a message that it's no longer presenting. Sorry, yeah. Right, so I talked about lasers now, and we looked at optics and fluidics both, and then how the cell and the laser light meet. The next question is, obviously I'm doing flow cytometry to collect a lot of different fluorescence, because if I'm looking at antigen 1, antigen 2, and all the way to antigen 10, I will tag all of these antigens to different fluorochromes. When I say different fluorochromes, I mean different wavelengths, and when I mean different wavelengths, I have to have different set of filters to collect all those wavelengths. Now, standard flow cytometers will have these three filters, most cytometers. Okay, let's study the first one. Long pass. What do we mean by long pass? A long pass filter, the job, as the name indicates, if you cut the word into two, pass longer wavelength. Look at the rating of this filter. It is LP500, which means long pass 500 nanometer. The job is anything greater or anything longer than 500 nanometer should be allowed to pass through the filter and reach where? Obviously the detector. Okay, I repeat, light greater than 500 nanometer is allowed to pass through the filter and reach the detector. What happens to light less than 500 nanometer? It is either blocked or reflected. The exact opposite of a long pass would be your short pass filter, where light lesser than a certain wavelength. For instance, in this case, it is a short pass 500 nanometer, light lesser than 500. Please mute. I repeat, light less than short pass 500 nanometer will be allowed to pass through and reach the detector. So this is the exact opposite of the first filter. If you cut the word into two, you will get the answer. Pass longer wavelength, pass shorter wavelength. Now the question is, you passed a certain longer wavelength and you passed a wavelength shorter than a certain wavelength. Now the question is, if I want to have a range, what do I do? Because fluorochromes always come in range. You will always have a range where they emit and where there is an emission maxima. That's when you use band pass. When I say band pass, I am passing only a range of light through the filter and reach the detector. For instance, in this case, the rating of this filter is 500 oblique or bar 50. Okay, a lot of people when they read these band pass filters get confused because of the way we write these things on flow, but I'll try to make it simple for you. 500 means the midpoint, okay, and 50 oblique 50 means the band of the light on the either side of the midpoint, which means it is plus 25 and minus 25. So if I subtract 25 from 500, it is 475. If I add 25 to 500, it is 525. So what's the difference? It is 50 nanometers. So that's the band of the light you're allowing to pass through. So whenever you read band pass filters, for instance 500 oblique 20, it means 500 plus minus 10, which means you're allowing light from 490 to 510 to pass through and reach the detector. So these are some of the most common filters people use in flow cytometry. There is something called dichroic also. Imagine the same filter. If I write it as DC, I repeat, DC LP 500, it means the filter can now split the light into two different wavelengths. One it will allow to pass through, the other it will either reflect to some other filter. So that's the job of a dichroic. Di means two, chroic means color. Split the wavelength into two. That's the answer. Now, every instrument, whether regardless of what type you have in your facility, will have an optical layout. Go through their manual and perhaps take a printout of the optical layout and stick it nearby the cytometer, so that it's handy whenever you design your experiments. You should know what filters you have and what detectors do you have for you to be able to buy a reagent accordingly. Okay, now a laser light is hitting the flow cell. This is where the cells are being passed. They interact with each other, and then in the direction of the laser light, I told you forward scatter gives you size. 90-degree to the incident light is what I said, side scatter and fluorescence. Now all these different emissions are really traveling together now. For instance, the green, yellow, red and violet, they're all traveling and they hit a filter called 550 DCSP. What does that mean? 550 dichroic short pass. So the fact that it is a short pass, what does it do? It will allow light less than 550 to pass through and reach this detector number 1 designated as FL1 here. Okay, I repeat, all the emissions travel together and they see this filter, and this filter, the only job of this filter is to allow light less than 550 to reach this detector. What does it do to light greater than 550? I told you this is a dichroic, which means it can split the light into two. Light greater than 550 will be reflected and it will go to another filter. What's the rating of this filter? It is 595 DCSP, meaning 595 dichroic short pass. Again a short pass. So what does this filter do? Allow light less than 595 to pass through and reach detector number 2. Okay, and light greater than 590 will again be reflected and it will go and see another filter. Okay, likewise, till you collect all the emissions and they'll reach all the different designated detectors. This is how an optical bench or an optical layout is arranged on every flow cytometer. The one that I showed you is from one of the Beckman Coulter machines called Navios. This is a CVIVD clinical machine rather. Okay, so this is how you read a simple optical layout. Likewise, you will have different optical layouts in different instruments. Please go through the manual, take a printout and try and read it. These are some of the standard lasers and standard fluorochromes, like even Professor Kumaresan pointed out in his session. FIT-C and all the way to, let's say, violet 610. Okay, FIT-C and PE are some of the most popular flow cytometry fluorochrome couples, as I say. And then you also have propydium iodide used for cell cycle or most DNA ploidy experiments. And then you have something called tandems. Tandems are basically two fluorochromes together. Okay. I'll talk about tandems slightly in more detail tomorrow when I talk about how to do multi-cannon panel design. So if you talk about 488 nanometer, these are some of the fluorochromes available of 488 nanometer. Why should I know that? Like I pointed out in the beginning of the lecture, if you do not know the electromagnetic spectrum, what is the excitation wavelength and what is the emission wavelength, you will never know what laser in your machine will be able to detect that fluorochrome. And the second question is, do you have that laser in your system? Okay, so always instrument configuration should be printed out handy in your lab, and then whenever you buy a reagent, you should match both of them and see if this reagent or this fluorochrome can be used on the machine that I have. Okay, imagine you bought an APC reagent which requires a red laser and your facility only has blue laser. You will never be able to do that experiment because APC requires red laser and you don't have one in your facility. That's why knowing this is very, very important from a practical standpoint. We talked about the laser light hitting the flow cell, and then a lot of emissions are collected. Different filters will do different jobs basis the wavelength, the incoming wavelength, and basis the rating of the filter. Where do they all go to eventually? They'll all have to go to the photodiode, because remember, fluorescence is a very, very weak signal. It's a very faint signal happening in nanoseconds. Rather, that's the time scale for fluorescence. So you have to amplify the weak signal. The only way I can amplify is if I give it to a photon amplification machine or photo detectors, where every photon is converted into voltage or current. Okay, this is what I mean by amplification. So the standard type of detectors that we use in flow cytometries are photodiode, generally used to detect forward scatter size. The other standard detector that we use is,
sorry to interrupt you.
yes, please.
Actually the screen is not visible for quite some time, for almost 5 minutes.
Okay, is it so with everyone?
Yeah.
Why did somebody not point out? Okay, is it visible now?
No.
Okay, sorry about that. Let me reshare again.
Yeah.
Is it better now? Hello?
It is presenting. Yeah, now it's okay. No,
So I think some of
anyone in the participant list they view?
Some of you would have missed this laser and fluorochrome chart. I was just trying to tell you these are different kind of fluorochromes available from different lasers, and these are the different filters that we use in a standard basis in flow cytometry. Okay, and I was talking about detectors right now. Photodiode is what we use generally to detect size, which is forward scatter. Photon multiplication tube, the traditional detection systems for fluorescence and side scatter. We have a new variety or a new type of detector which will combine the best of both worlds, the best of photodiode and the best of PMT, and give you much higher amplification or much better sensitivity, is the avalanche photodiode. Okay, essentially it's a modified photodiode, much more advanced. Okay, now these are the standard photodiodes or standard detectors we use on flow cytometer. On our Cytoflex, on our even our DXFlex range of flow cytometers, we only use APDs, avalanche photodiodes, to detect both forward and side scatter, because they are the most advanced form of detectors available right now in the market, as opposed to PMTs. Okay, now there's a word that I have included here just to make sure that people understand this concept. It's called quantum efficiency. In simple terms, it's the ability of a detector to convert photons into electrons. Okay, how many photons in and how many electrons out. If one photon in, can I generate 100 or can I generate 50 electrons? That's what the efficiency of a photodetector is. That's what we mean by quantum efficiency. If you look at the graph on the right side here, the red line indicates the quantum efficiency of the PMT based systems. Okay, look at what happens when the PMT signal reaches 600 nanometer. Okay, it will start dying, which means the quantum efficiency decreases as the PMT starts seeing the red zone or the red spectrum. Okay, this is because of the property or because of the way the PMTs are built. Okay, now look at the APDs. I mean, this is a third party article comparing both PMTs and APDs. If you operate with an APD, because of the higher quantum efficiency, it has a good spectral range all the way from 400 nanometer to all the way to 1000 nanometer. Okay, which means the quantum efficiency is almost constant whether it is at 400 or at 1000 nanometer. This is the reason why APDs are much more advanced than PMTs. So this is the third and the most common type of available detector even in today's time. I'm sure some other manufacturers have this on their machines, and even microscope manufacturers. Now this is detectors, but you and I still don't understand this language because all of this is analog signal. It is a voltage pulse. On the x-axis you have intensity, on the x-axis you have your time, and on the y-axis you have your signal intensity, which is voltage basically. You and I still don't understand this. Okay, so how do I convert this? How do I digitize this? That's the job of an electronics in the flow cytometer. That's the job of an ADC, analog to digital converter. I pointed out in the beginning of my slide, if you have a small cell, you will have a small signal. Okay, let's say I have one small cell and then I populated somewhere at around 400, so I added one cell. I also detected one more small cell, so I keep on piling up all the cells and I bin them, I group them on detector number or channel number 400. Likewise, if I have a bigger cell, let's say tens of them or 20 of them flow together, I will place or bucket all of them under 800. Okay, so this is basically the signal intensity increasing from left to right. All small cells falling on 400, all big cells falling on channel number 800. Okay, now this is in the idealistic world. I mean, obviously in the realist world it won't happen that way, because biologically you will always have a distribution. No two cell types will be almost same. Okay, so therefore you'll always have a bell-shaped curve in your actual experiment. So this is what I mean by a histogram, and this is how you display data for dot plots. Okay, these are the standard displays that people use in flow cytometry. The basic take-home message of this slide is basically you have an analog signal which leaves the detector, which needs to be digitized and converted into a human readable format. And basis the size and basis the different aspects, it will be binned separately or it will be grouped separately, finally leading to display. I'll play a small animation. This is without the sound, but I'll keep talking just to summarize of what things that we have discussed so far. This is a flow cell and the laser. Now you have the cells flowing. Right. So this is all fluorescence and scatter. You put your sample first. Imagine a 5 ml tube. By suction, by pressure, by pressurization, it starts aspirating and then it starts shooting it to the laser beam. So as and when the cell starts interacting with the laser beam, in the direction of the light is forward scatter. 90-degree was what I mean by side scatter and fluorescence. And remember, this is a cone of light we are collecting. We are blocking the central laser beam because that's very intense. If you allow it to hit to the laser, the detector will be damaged on day one. Now, this is how the cell and the laser light are interacting. This is forward scatter. This is one size of a cell. Perhaps in the next few seconds, we will also look at how, if it's a different size of the cell, where the intensity varies. Look at this, small size, slightly bigger, and much bigger. So therefore you have proportionately different scatter signal, like I pointed out in the beginning of my session. So this is still a voltage pulse. If you have a small cell, it will all have a small pulse. If you have a bigger cell, it will give you a bigger pulse. Okay, this is called a voltage pulse, because essentially, remember, photons are converted into voltage in the detectors. That's why we call it as voltage pulse. Now you have forward scatter and then you have side scatter and fluorescence to the 90-degree. This is how side scatter would be measured. It will measure all the internal components of a cell type. Okay, a small cell, slightly bigger, even bigger. So lympho, mono, and granulocytes, different sizes of voltage pulses basis the different properties of the cells. Okay. Now, if you combine two histograms together, I'll talk about histograms in greater detail in the next 1 or 2 slides. You get a dot plot. These are the standard displays on a flow cytometer. Okay, a lymphocyte, a granulocyte, and a monocytes on a forward versus side scatter. Okay. Now, we looked at dot plots. In fact, when I talked about FSC versus SSC and I showed you RBC, WBC populations, I talked about dot plot. The first plot where I'm pointing right now, let me see if I can use a different, no, I can't. All right. So the first thing where I talked about different sizes of these blood samples, look at this. This is called a dot plot. Okay. Now, why is it called a dot plot? I mean, the name is misleading. It's not called dot plot because you see dots on the plot. Essentially, it is called a dot plot because you can measure two signals at a time, one on X, the other on Y, which means I am measuring two signals simultaneously. One could be forward scatter on the X in this case and side scatter on the Y in this case. Okay, you can measure any two of those signals together. Essentially, it's a bivariate plot. That's the definition of a dot plot. Now all the things to the right of this plot, the density plot, the pseudo color, the contour, they are just different representations of the same data. For example, if you look at the first plot and try and distinguish, let's say, monocytes from granulocytes here, very difficult. I mean, unless you are a very experienced user, it looks like both of them are same. Okay, or maybe perhaps you will include a box here and then call them as monocytes, even in reality they could be some of your granulocytes as well. So how do I get a better visual or a better representation of this? Go straight to the right, look at the contour plot, the place where the contour ends. It's like, you know, taking a peak and chopping it into multiple pieces like an onion peeling. You will see contour representing the same data in a much better way. Okay, so always for better quality publication or better presentations, use contour plots than your regular dot plot. Okay, and this is just a colored contour, the pseudo color, or this is just a grayed or a gray color or a black painted contour. All of them are the same thing. It is just the way you present the data. Now this is a dot plot. If you use only one axis for measurement, which is x-axis mostly, that is your histogram. And I told you how to read histograms. The intensity will increase from left to right, which means this is a negative peak. And as you start moving to the right, the small peak is a positive peak for CD4 in this case. Okay. This one is an overlay. Overlay means you can superimpose multiple histograms of this kind and then get an overlay. For instance, if you're doing drug studies, if you're looking at different doses of drug concentrations, the effect of that on your cell cycle, let's say, or for instance, the cell health, let's say, you can superimpose all of them and then study using overlays. Likewise, you can also use dot plot overlays, which is much more interesting than your histogram regular overlays. So these are the different data displays. I mean, general, the most available on all cytometers data displays in flow cytometry. Dot plot, remember, is a bivariate plot. Histogram is univariate, which means you're only measuring one signal at a time and mostly x-axis. The y-axis is always count. Now, this is one area where a lot of people get confused as to what do you mean by region and what do you mean by gate. Remember, I told you I have a blood sample and I don't want to look at RBCs. Perhaps even I don't want to look at monocytes and granulocytes. I only want to work with lymphocytes here. How do I select only that population? On your software, on any of the machines that you have, you will get all these different shapes. Okay. In this case, I have taken a free polygon shape. Make a boundary around the population of your interest. In my case here, lymphocytes. Okay. This is called drawing a region. I repeat, when you select a population or identify a population on your data plot, scatter plot, that's called drawing a region. Okay, this is not gate. When you apply that region to the second plot or perhaps third plot, I repeat, when you apply the selected population onto the second and the third subsequent plots, this process is called gating. Okay, and the plots that had this population would be called gated. Okay, this is what I mean by region and gates. Gates can be a combination of many different regions. I can take lymphocytes, monocytes, granulocytes, call them gate region 1, region 2, region 3, combine all the regions and then call it as gate 1. Okay, now I took out lymphocytes from the first plot, took them to the second one, and then now I'm looking at how many of them are CD3 positive in green and CD3 negative in purple here. Okay, so, and then I work with, let's say, CD3 negative cells, take them to the next plot and see how many of them are B cells and how many of them are natural killer cells. Okay, because 56 plus 16, that marker is for NK cell, CD19 is expressed on B cells. Okay, you will get used to this if you're not an immunologist or if you don't study immunology regularly. You'll get used to this as and when you start studying a lot of papers, so don't worry. The idea here is when you select a population, that's region, and when you apply that population to the second and the subsequent plots, the process is called gating. I hope that's clear right now. The other concept people confuse themselves with is voltage and gain. Remember, I told you when a particular signal goes to the detector, it is still a voltage pulse. Okay, not necessarily always it will have a bright signal or something that I can say very distinct from the background noise or the instrument noise. How do I amplify signal? Only if I make available more current. Okay, the only way that I do is on a flow cytometer is apply voltage and gain. Some instruments give you the option of both voltage and gain. Some only operate only on gain, like the much advanced APDs. Take an example of the extreme left. The population was inside the axis, which means not enough amplification at gain 1. Gain 1 meaning this is just an arbitrary value on the software. The peak on the right, it's a positive. The peak on the left is negative, but then I don't see great distinction between the two of them at gain 1. The moment I dial up or increase my gain value to 2, what was inside in the first plot, all the negative population now is coming out. It is sticking out now, which means the amplification may be good enough. Okay. Now, the moment I change the gain value from 2 to 10 or voltage value from 2 to 10, both the population starts traveling to the right. If you keep on doing this, you will not see any red signal going forward, which means you will not see this histogram or this peak at all. Why? Because the amplification is too much. This is called detector saturation. Okay, basically you are overeating and therefore you can't eat any further. So there will be a point where it will be optimal, where you see both negative and positive peaks on the data plot. That's when you have to stop adjusting voltage and gain. The last bit of information before I end this presentation and I go step into immunophenotyping is the concept of threshold. Remember, I told you a lot of your sample preparation, whether it is your cell lines from incubations or for that matter, your blood cells, if you have lysed RBCs, no God on this planet can ensure 100% RBC lysis. There will still be a lot of RBC and broken cells left in your sample tube. So this is what I define as debris or unwanted, as some of the people say. Okay, that's not right. Actually, it's simply to say debris. Look at the first plot. I am now setting the threshold. Threshold means the minimum at which the signal starts to rise or the signal starts to be recorded by the electronic box. It is no longer presenting. Is the screen visible? Still visible. Okay, good. Now, if you look at the first plot, I am thresholding it somewhere here. Let's say less than 256 number. Okay, and I'm chopping some of the RBCs. My lymphocytes are still good. My monocytes are still good. My granulocytes are still okay. Go to the second plot. I have increased my threshold some bit. Okay, I see a lot of white space here. I'm moving from left to right. In fact, I think I have cut some of my lymphocytes also, but I'm not sure. Okay, but I'm still okay. I still see lymphocytes, monocytes, granulocytes, the standard pattern. Look at the third plot, C. If I keep increasing this from left to right, now I have excluded all my lymphocytes also. Perhaps that those were my population of interest, but because I didn't do my threshold properly, I overdid it rather. I have now chopped off my lymphocytes as well. When I say chopped off, what do I mean? I am basically instructing the system or basically instructing the electronics box not to look at these cells at all, which means it won't record. This data is lost forever. Okay, so threshold or trigger or discriminator, as it is called in some other systems, is a double-edged sword. Use it very judiciously and very, very carefully. Okay, if you do it good and well, I mean, you will have all the population. If you overdo it, then you won't have any of your data available for further analysis because the electronic box is not recording it at all. Okay, now finally, when you do all of these things, obviously you will have some data output and you got to look at some statistical data, some statistical estimations. The key questions you should ask yourself as a biologist or an experimental immunologist, let's say, is what kind of data are you looking for? What kind of fluorescence are you looking for? If you're looking for, let's say, GFP fluorescence, how much fluorescence do I have? How much fluorescence is Y greater than X? What percentages of cells are positive for my marker? What percentage of my cells are GFP positive? What percentage of them are actually transfected to signify transfection efficiency? These are some of the standard questions you should ask yourself before you begin your data analysis. Some of the common stats available on any flow cytometer in today's time is the mean, median, mode, the percentage CV, geometric mean or arithmetic mean, the percentages of different populations. All of these things are available on a standard basis on most cytometers. So technically, you can take this data and then do your statistical analysis. Remember, but the data is only as good as your input was. If your sample preparation was not good, if the cells were not treated properly, if the fluorochrome was not added properly, if there were mistakes in weighing, if there were mistakes in pH, if there were mistakes, let's say, in pipetting, all of them will reflect in your data. So the basic science, the basic chemistry that you do on a day-to-day basis in your lab is still very much important. This is what we mean by pre-analytical errors. Okay, so no amount of data analysis technique can make a data look good if the input itself was bad. That's the Shapiro's seventh law of flow cytometry. Shapiro's book is considered to be the Bible on flow cytometry reading. Now, I talked about a physics, I talked about a bit of biology. How do they all come together? Let's take it in a basic biology workflow. Take a blood sample, for example. I don't need RBCs, so I lyse RBCs. I label with it an antibody. Now, the antigens are in your blood sample. The antibody that you add will interact with the antigens. They make an antigen-antibody complex, and then you pass them through a flow cytometer. The laser will hit these cells, the interaction happens, and then you have different wavelengths collected. Different filters will collect different wavelengths. Eventually they are all sent out to detectors, and then finally data display. This is how I just simply wanted to translate you from a physical world, I mean rather the world of physics, to biology. This is what you do on an experimental basis. Okay, that's the end of the session 1. Before I begin immunophenotyping, if you have any questions, please do ask. Kumaresan sir, over to you.
So fine, sir. It's a fantastic lecture. I leave to the participants, let them ask the questions.
Should I look at the chat box or something? Okay.
Yeah. I think we also have the feedback forms. They also can type there, and then we can answer later also. That's not a problem.
Okay, the questions are, please explain the type of plots in data display slide. Okay, I'll do that. And then you have major limitations. Okay, the major limitations of flow is you have to have cells in single suspension, which means if you have clumpy cells or if you have clumpy cell lines, the flow cell will get choked. Remember, the average opening or the diameter of a flow cell, the inner diameter of a flow cell is 5 to 7 times bigger than the average lymphocyte. For instance, if your average cell size is 10 micron, my flow cell opening would be roughly about 70 microns, 5 to 7 times bigger. Okay, anything more than 70 micron will end up choking the flow cell. So it is absolutely essential that you have cells in single cell suspension. That's one of the limitation, I would say, apart from the fact that you cannot do localization studies on flow. You cannot tell me where exactly the protein is sitting inside the cell, whether it is on, whether it is inside. They're all indirect ways that we do on flows. Okay, so that's the major limitation. Types of plot. Okay, let me see if I have types of plots. There you go. Okay, the one that I'm pointing out right now is a dot plot. Why do I call it as a dot plot? Because it's essentially measuring 2 signals at a time, one on the X and one on the Y. So it's a bivariate plot. I'm measuring forward scatter on X, I'm measuring side scatter on Y. So size and granularity can be both looked at the same time. This is the advantage of a dot plot. You're looking at interaction of 2 different signals together. This is a histogram. It looks like a bar graph, your regular Excel bar graph, where you're only measuring signal on X-axis here. On the Y-axis, it's always the count, the number of these particles that you are measuring. Okay, so this is a histogram. The same, these two are the standard default plots available on all flow cytometers, except I think 1 or 2, which works on a different principle. Now, dot plot, the same dot plot, I can represent it in multiple ways. One of the best ways you can represent your data, flow cytometry data, when it comes to dot plots is contour. Because if you look at the last plot here, the demarcation is very clear from a lympho, mono, and a granulo. You will know exactly where to draw your regions, as opposed to the first plot where it is still a little smudgy. Okay, so that's the advantage of contour plot, but essentially contour is also a different way of representing a dot plot. There are only standard 2 types, remember, a dot plot and a histogram. I hope that answers the question. Any other questions, please provide. Okay, that's okay. And please explain the gating part. Can it differentiate 2 gram negative bacterial rod cells? Yes, if you have staining available, people do that. will have different effect. Sir, will UV laser and visible laser have effect on the cell? No. The amount of time that a laser or a cell spends with the laser beam is in microseconds, roughly about 6 to 10 microseconds. So it's very, very less for a laser light to do any effect or have any effect on the cell type. And besides, remember, I am only taking a cone of the light and you're making it interact with the cell. I'm not really hitting the actual laser beam, the central intense laser beam to the cell. Okay, and that's why there is no effect. I hope that answers your question. There was one more question for gating. That's the next slide, I believe. Okay, I hope the slides are visible. If not, let me know, please. Now, this is the first plot where I have my lymphocytes, granulocytes, and monocytes. I have my lymphocytes here. Now, I only want to study lymphocytes and I'm not interested in other populations. So how do I select this? I select this by using any of the shapes available on a software. This could be a oval, this could be a circle, this could be a square, this could be a polygonal, depends on the options available on all different kinds of softwares in the field. Okay, so in this case, I have taken a polygon free shape. I draw a boundary around these lymphocytes, the one in red. Okay, and then take them to the second plot. The process of applying this region to the second plot or even third and fourth plot is called gating. Okay, I repeat, when you draw a boundary and you select a population, that's region. When you apply that region onto the second and third plots, that's called gating. The second plot is now gated on lymphocytes, which means the second plot, the display is only because of lymphocytes and it doesn't have any contamination from either monos or granulose. How do I know that? Because I also have a specific marker here. Okay, that's the definition of region and gates. Somebody says when cell sorting and gating is needed to be done. I'm not clear with your question, because whatever you do on flow cytometer, gating is essential to identify your populations, and then you can send them for sorting or send them for different analysis. Okay, Shubham. Murli Ramchandran, voltage gain will increase both the analyte signal as well as the noise. So when the gain can be used effectively? Good question. Now, this is called a voltage walk or a gain walk or a titration simply. Okay, I keep on increasing voltage. At some point you will get a signal where the positive doesn't move any further and the negative stays wherever it is. Okay, there is a voltage point where you have both negative and positive staying still. Okay, that's the ideal point of voltage and gain. This is what we mean by voltration or voltage walk. So you have to do this for the first time before you set your experiment. Only then you will be able to differentiate signal due to analyte and background noise. Okay, noise cannot amplify after a while. No matter what voltage you apply, the noise will stay wherever it is. You will see on the cytometer that your events per second will go up rapidly. Initially, let's say it was 1000 events per second when you look at the flow rate, but now because the noise has increased so much, it will go to 10,000 and 20,000. That's how you identify high noise. Okay, so you have to come to a point where the noise is not being amplified, the signal is also staying still. That's the best point. Does providing charge to the cell surface is also a way to use sort cells in flow cytometer? I mean, yes. I mean, the only way you can sort is through the electrostatic sorting. I mean, there are other ways that people purify cell populations. There is magnetic beads people use for sorting cells. Then there is electrostatic sorting, which is the standard, one of the most popular way of sorting cells. And then there are these regular columns from, I think, Kyogen or some other company which use negative and positive selection methods to purify different cells. Okay, but if you use electrostatic sorting, this is a charge-based sorter. You apply charge, you give differential charging to different cells and then send them to different vesicles. I think we don't have any other questions. Where does lymphocytes produce, sir? I didn't get your questions, Sidhu Dudgi. Where does lymphocyte produced, as in the human body? I mean, all of them have to start from bone marrow, and eventually these T cells, for example, they mature in thymus. If that's the question. Okay, and yeah. Shall we take a 2-minute break or we continue with the second session, sir?
We'll take 5 minutes break, sir. Fine.
No problem. Till then, if there are some questions, let me take a look at it. Can you explain why is it necessary to sometimes apply high pressure to the sample while setting them in flowing motion. Okay. See, sometimes when you have very little sample, for instance, the regular flow cytometry we do about 1 million, or let's say for example, now let's say you don't have 1 million, you have only 0.1 million, which means the cell number is a limiting factor. You don't have enough cells. If you have very low cell number and you operate on low flow rate, my friend, you will spend the entire day in front of the cytometer just to acquire one sample. As long as there is sample in the tube, it will keep acquiring and you will not get the required statistical output. So it's just to speed up things, you get different flow rates or different pressures, as I say. Okay, hope that answers the question. Can you play the animated video again? Yes, I can. Here you go. Others feel free to take a break, and I'll get some water for myself. So that's a voltage pulse. Depending on the chemical and the physical properties of the cells, you will get a different voltage pulse. Remember, when I say 10,000 cells per second, I'm actually measuring 10,000 voltage pulses. What you see finally on the flow cytometry data plot or histogram is a aggregate value of such 10,000 voltage pulses. Every cell or every event that passes through the laser beam will generate an voltage pulse. Okay, so that's the animation for you. If there are no further questions, I will go to immunophenotyping. We'll wait for another minute for people to come back. Link is not working. What do you mean by link? Or the feedback form. All right, thank you.
Sir, once I have a question. Actually,
yeah,
the analysis probably happens within few milliseconds. So within that, the sorting also happens. Or if can you just tell a few words about that?
I mean, sorting timings? Essentially is, I mean, when you pass your cells through the system, I mean, let's say it spends a certain amount of time, usually microseconds in the flow cell. That's called dwell time. Okay, you can vary this dwell time or the amount of time that the cell spends in the laser beam on a sorter, because on a sorter you can vary sheath pressure. Remember, on an analyzer everything is fixed. It's generally 4 psi, but on a sorter you can go from 10 psi, 30 psi to all the way to let's say 80 or 90 psi. So by altering the sheath pressure, you can increase or decrease the dwell time. Okay, the time that it spends in the laser beam, and then do your sorting. The next question is, once you select this for sorting, and then there has to be an immediate charging and decharging mechanism. That's what you do. You use an electric circuitry, apply charge onto your selected populations, and then send them to different reception collection vesicles, basically. Okay, so the answer is dwell time can be increased or decreased using different pressures on a sorter. I mean, was that your question or anything else?
Yes, exactly.
Thank you. Some of them say the link is not working, sir. The feedback form, I think.
Yeah, that will take care, sir. Fine.
Sure. What to do if the flow channel will get clumped? Good question. Okay. Now see, I mean, the flow, I mean, it's not flow channel, it's actually flow cell or flow chamber. Okay. Channel is only when you talk about detectors on a flow cytometer. That's the distinction. Now, when a flow chamber or a flow cell gets blocked, it could be many reasons. I mean, it could be that the air is stuck there because you sample, there was no sample in your tube and you kept on acquiring, because the flow cytometer does not understand this. It kept aspirating air. Okay, therefore, it's not moving any further, or perhaps too much cell concentration or too much of a bigger cells, they're all accumulated at the neck of the flow cell. It is now choked. You have a lot of different maintenance programs on most cytometers in today's time, but the most standard that anybody can do in most labs is run a solution of bleach, mild lab bleach. Okay, usually 1% sodium hypochlorite or 0.5% sodium hypochlorite is what people run, and then you have commercially available detergent bleach. Run them for about 5 minutes. It will take away all that is choked, and then followed by let's say a 5 ml 5 minutes distilled water run as well. So for about 10 to 15 minutes, if you keep cleaning, most of the issues should be taken care of. Okay. The other thing is, from a sample preparation point of view, you have to take care that it is not clumping very badly, that 90% of the cells are still in single suspension. Okay, perhaps you can use a nylon mesh and then filter it through a 30 micron nylon filter, and then give it to the flow cytometer. There are other aggressive forms of cleaning called drain flow cell, or perhaps you can use boost pressure and then apply a lot of pressure and then drain all the contents of flow cell. That way also you will know whether your flow cell is now cleaned. The best way to understand whether what you have done is working or not is to run those quality control beats. Okay, every instrument will give you quality control beats. Run them. Now, if your beats are being seen by the laser and you see detection on your data plots, which means the flow cell is clean, which means the clog is cleared. Okay, so that's the indication. Don't try it with your cells. I try them with your beats. I hope that answers your question. The basic answer is clean it, clean it, clean it. How to read the graph representing differentiated cells based on markers, say 4 and 8. I'll come to that a little later, Babita, when I talk about immunophenotyping. Stay with me till then. How is the sensitivity of a flow defined? What criteria do we consider to define the same? Very good question. All right. See, sensitivity of the flow is the minimum amount of particles it can detect. Let's say the minimum amount of dim particles it can detect from noise. Can I detect 10 molecules or can I detect 20 molecules? If I throw 10 molecules of FITC at a cytometer, will it be able to detect this very low concentration of FITC from noise? Okay, that's what I mean by sensitivity of a flow cytometer. Generally, they run rainbow 8-peak beats and then check the sensitivity of all the cytometers. If you look at all the manufacturers, when they talk about sensitivity, they talk about running these 8-peak beats, and then they take the lowest peak, which is the dim peak, and see how well it is separated from the noise. And the other question is the detection limit. How many of them can I detect? If I only have 10 antigens, or if in other words, I have only 10 fluorochromes, will my cytometer still be able to do a good job? That's what we mean by sensitivity. Resolution, when I talk about, it's about how narrow or how broad the peak is. Okay, that's resolution. I'll talk about this in great detail tomorrow when I talk about multicolor flow cytometry. I have a slide exclusively on sensitivity and resolution. Babita, don't worry. Thanks for asking that question. I'm not sorry, this was not Babita, this was Smritishri Mohanty. Thank you for that. Right now. All right, shall we begin with the next session? Somebody can unmute and let me know if the screen is still visible. I keep getting this is not visible. Babu, you're there? Is the screen visible?
Yes, sir, it's visible.
Thank you very much. Right. Discussed about the basics aspects of a flow cytometer, the basic principles of flow cytometer. One thing before I begin my immunophenotyping session, I would want to clarify is a lot of people call or used to calling this as fax. Remember, you call this as a fax only when you're sorting. Okay, flow cytometers are available even without the sorting function, and they are the most popular ones that you see in most labs across the world. A machine which cannot sort is called an analyzer. A machine which can sort is called a fax, and only that machine should be called as fax. Not every flow cytometer is a fax machine. Okay, remember this. Right. With that, let's go to the immunophenotyping. Now, we talked about how different components come together and interact with each other. Now, how if I add different antigens, different antibodies to various antigens on the cell type, I can classify them as a T cell or a B cell. Let's study that in great detail. What is immunophenotyping? Basically, you're classifying different cell types of the immune system. Okay, I repeat, you're classifying or you're phenotyping. Okay, you're differentiating one cell from the other of the immune cell type based on what? Based on the expression of a protein on its surface, or perhaps some protein expression within the cytoplasm, or you could also take a look at nucleic acids themselves. Okay, using any of these properties together, put them on a dot plot, for instance X and Y, you can distinguish two different cell types from one another. That's the essence of immunophenotyping.
So can you pause for a second? Actually, the slides are not visible.
Okay, thank you. Let me share that again. I will present that again. Let's see if it's there you go. Are they visible now?
Yes, sir. So actually, the initial page was showing up. Now it's visible. Thank you.
Okay, thank you. I mean, it might happen for some who have low internet connectivity, and for some who have, issues with internet connectivity, it might happen. So let me know as and when you don't see the vision. Thank you. Now, that's the outline of what we are going to discuss in the next, let's say, 40 minutes before I open it up for questions again. We are going to talk about briefly a phenomena called fluorescence spillover, spectral spillover as it is also called, or bleed through as it is also called, and how do I mathematically correct this? And then a little bit of basics of immunophenotyping. I mean, do I do immunophenotyping only using flow, or are there any other methods? Why is flow immunophenotyping advantageous, etc.? I am briefly going to talk about what antibody titration and stain index is, although we will discuss this in great detail in the session tomorrow. I'll skip this for today. We will discuss a basic protocol so that for some of my postgraduate friends who are on the call today, this will be much more useful, because biologically, how do you actually run this experiment in a lab, let's say, is what we're going to discuss here. And then I'll give you an example of a multi-antigen panel or a group of antigen experiment, what we call as panel. We talked about different filters, different fluorochromes, different wavelengths. The fact that there are different filters, different fluorochromes, different wavelengths obviously means they are all going to talk to each other, which means they are going to be seen by each other in each other's detectors as well. So this is what I mean by spillover. For example, what I have on the display right now here is a band pass filter. When I've made a boundary here, all of them mean band pass filter with the rating as well. 5, 25, 40 means 525 plus minus 20. Okay. Now, when I excite all these dyes, FITC, PE, PerCP, and PE size 7 from the same laser, I repeat, when I excite all these fluorochromes from the same laser using these kind of band filters, let's see what happens. The FITC signal starts to rise, the maximum is collected in the FITC house, but does it stop within the band pass? The answer is no. It also continues to travel and then being seen by other detectors. Now imagine this detector was meant for PE, but then I see some bit of this green signal also in the yellow orange. Okay, this is what I mean by spillover, spilling of one signal into another. Okay, why does this happen? Spillover, remember, is an intrinsic property of the fluorochrome itself. The fluorescence spillover is an intrinsic property of the fluorochrome. By nature, in nature, they have long tail emission, which means their emission peak is not just one midpoint. They always carry it through a certain wavelength and then eventually die. Okay. For example, in the case of FITC, I have collected the maximum at around 530, but then it still keeps traveling all the way. I can still see a very faint green band all the way up to 700 nanometer. Okay, although very, very little after 600 nanometer, but at least it is causing a lot of problems till 600. But the emission of PE is somewhere in this range, so there is an overlap. Now imagine if you are studying two different antigens, okay, antigen 1 on detector 1, antigen 2 on detector 2. Now how do you know whether the signal is from antigen 2 alone and not a contamination from the first one? Okay, the only way you would know and the only way you would get rid of this contamination is something called compensation. It's also called spectral correction or spectral compensation. Likewise, if you see FITC will all the way travel to, let's say, even some bit of that in the third detector, but then in the fourth detector, very, very little of FITC. In fact, I think it will be nil, almost 0.000% in the detector fourth. What does this tell you? This tells you one fact, that if two fluorochromes are sitting close to each other, spectrally, if they are close to each other in terms of their emission profile, I repeat, if their emission profile is spectrally close to each other, let's say in the order of 40 nanometer difference between two, they will have spillover. If they are far apart in terms of spectral range, you will not have issues of this spillover. You will not have any bleed through. That's the take home message of this slide 1. I hope spillover concept is clear. Likewise, if you take PE, I excite PE with the same blue laser, it starts traveling from here, which means some of the PE signal is also seen in detector 1. Okay, although it's very, very faint and very, very little, but still it's a contamination. We have to get rid of that. Okay, but then because I told you the fluorochrome will always have a more tailing on the right side, look at what PE does to per-CP here, what PE is doing to detector 3. Okay, look at how it is traveling and causing contamination in this particular detector. Okay, so the idea is, if they are spectrally close to each other in terms of their emission range, you will have a lot of spillover and you have to correct it using a process called compensation. Let's understand from a practical point of view, how do you do a simple two-color experiment? Okay, now some of you asked, how do I distinguish different cell types based on different markers? This could be one of your example as well. I am taking human blood sample here and I'm trying to study lymphocytes, in particular CD4 T helper cells and CD8 T cytotoxic cells. I have tagged 4 with FITC, 8 with PE, which means there are 2 different fluorochromes and you will have issues with spillover. By default, as the first step when you set up your compensation experiment, you have to run something called unstained, which means unlabeled tube. Just take the cells, chop the RBCs perhaps, and then give it to the flow cytometer and look for something called autofluorescence. What do I mean by autofluorescence? Fluorescence by itself, the cellular fluorescence itself without any reagents. Why does this happen? Okay, there are a lot of flavoproteins within your cytoplasm of a cell. They will pick up the laser light and start dancing around. That's why you see autofluorescence. Okay, but fortunately this autofluorescence is only 1 log usually on the scale, or perhaps 1.5 2 logs. Okay, it's not too wide so that it occupies the entire plot. But for some cells, especially when you're working with large cells like, let's say, mesenchymal stem cells, they have extremely high autofluorescence. For instance, your neutrophils have very high autofluorescence when you compare this with lymphocytes. So different cell types will have different levels of autofluorescence and also different conditions. For example, if you draw fresh blood and look at lymphocytes, the level of autofluorescence will be different versus lymphocytes which were cryopreserved or lymphocytes which were stored. Okay. So there will always be an autofluorescent difference. Always use the same type of cells for your experiments and same conditions. Tube 1, run unstained, look for autofluorescence, and then tube 2. Now you have shown the system how autofluorescence looks like without any reagent addition. The next step is show it how color 1 looks like. Color 1 in our case here is FITC. Always do compensation from shorter to longer wavelength, increasing order of wavelength. Okay, so in this case FITC. Take FITC alone tube, which means I have added only antibody of CD4 FITC, run through the cytometer, okay, and then you should see obviously FITC signal, but you will also see a little bit of FITC traveling to PE. How does that happen? We'll go to the next slide and see that. Tube 3 should be the second color, which means now the system knows how to identify FITC only. Has it learned how to identify PE only? Not yet. So run tube 3, show it how PE only looks like. So you have now shown 3 tubes to the system. One, how unlabeled look like, and then the color 1 and the color 2. Finally, when you throw a mix of all of them together, it's like teaching a child how to identify a banana from an apple. I show it a banana first and then an apple, and then mix all of them together and then present it, and then it will be able to identify a banana from an apple. Same thing for a flow cytometer. When you throw a mix right now, it will identify, let's say, FITC from PE. That's the tube number 4. Now, what if I have more colors and more fluorochrome? Some of you might be working with 3 colors, some of you might be working with even 20 colors. The logic still remains the same. The first tube will always be unstained. All that you will do is you will keep on increasing the additional single controls. All these things are called as single color controls because you are just displaying single color to the cytometer. Okay, more and more number of fluorochromes, keep adding the additional fluorochrome tube, display it to the cytometer, and then do the compensation. Okay, now this is how it will look like when you do it on a cytometer. So I run my unstained tube, and on my X I have FITC, on my Y I have PE here. Okay, and I told you the intensity will increase from left to right, bottom up, which means the first bit of things that I'm displaying right now is negative, dim, positive, bright positive, likewise from bottom up as well. So I take the instant sample, adjust the voltage and gain in such a way that my negative population in the first log. Okay, usually first log. This is the standard or the most common way people set up compensation autofluorescence. Okay, now is this correct? The answer may not be a yes always. There are better ways to do this because there is no statistical basis to say why should I place my population on only 10 power 3, why not below 10 power 3 or slightly above 10 power 3. There is no statistical basis to say that. The only logic is adjust this in such a way that you have enough space for positive samples. If you keep your unstained all the way at 10 power 6, where does your population of positive populate? It won't have any space left, correct? That's the reason why you put it in the first log. That's the first step. And then note down something called X median, the place where it is cutting X, and Y median of this population. Okay, medians are all available on the software. Before we go to the tube number 2, which is your single control, I told you autofluorescence is primarily because of the properties of the flavor proteins inside your cell type. But then if you also have neutrophils, they are naturally highly autofluorescent than, let's say, lymphocytes. And then if you have very high autofluorescence, for example, for some cell types, use very, very, very bright fluorochromes for these cell types to amplify the signal from the autofluorescence level, or put them in a detector where the level of autofluorescence is very low. For example, all your red channels, for instance, all your 650 nanometer and above, these are red channels. Put all your highly autofluorescent cell types using those channels, then you will still be doing fine. So just a pointer in terms of how do you set up your autofluorescence experiments if you have these kind of cell types. Now, you ran tube number 1, you adjusted the voltage on the X, on the Y in such a way that you have placed your population of interest on the first log. Okay, this first log could be different for different software, so don't worry, the scale does not matter here. What matters is the logic. Now, in the second tube, when I run my FITC only, remember, what I'm having in my tube is only FITC. This is the box of FITC. This is the house of FITC. But then why is my signal traveling all the way up? It is showing itself also in the PE detector. If I cut my PE detector, let's say 10 power 3, that's the negative. Anything beyond 10 power 3 on this plot is positive, right? So, but then my FITC had no job being seen in PE. Why does this happen? If you remember for the first slide, happens because there is a spillover from FITC to PE. Okay, now because there is spillover from FITC to PE, look at this. This is the unstained population. This is where it was supposed to be, but then now it is traveling upwards. What do? I have to bring it down. The hand tool that I've shown here is on the Cytoflex. All that you have to do is click on that hand bar and bring the population down. That's how easy compensation is on this machine. Okay, but you still have to do a manual confirmation on most machines to know whether you have done the compensation correctly or not. How do I know that? The only way to know that is match medians. This is called median alignment. Remember, We measured the X and Y median of the unstained. The right unstained, I repeat, the right unstained is the unstained of the stained tube. What do I mean by that? Every stained tube will also have some bit of unstained cells because they don't stain for that particular marker. In this case of CD4 FITC, I will have all CD8 NK cells, B cells as my negative. So they are the right negative. So we'll come back to this compensation again. I ran FITC only tube. FITC is spilling into PE, therefore it's traveling from bottom up and seen in the double quadrant. I bring it back. When do I stop is the next question. I stop when the median of the x-axis matches with the median of this population. If you look at the y median of unlabeled, it's at about 450, and the FITC median that I just adjusted is at about 460. I'm sorry, the FITC population is at 460. Both of them are very close to each other. That's when I stop adjusting my compensation. You can always keep doing this and then you will go down further or even further, but then the only way to know whether your compensation is right is by matching median. And what's the formula? Always ask two questions. Where is the spillover coming from and where is it resulting to? In this case, the spillover is resulting from FITC. It's coming from FITC and it is going to PE, which means the problem is happening in PE detector. Okay, so PE will come first in the equation. PE minus a portion of FITC signal that was seen in the PE detector, which means percentage FITC. That's the formula for compensation that you will use to do this. Okay, on most modern day instruments you don't have to really look at the formula. They give you compensation matrix like this, and if you understand where is the spillover coming from and where is it going to, you will simply use a toolbar like this, bring the population down, place it here, match the medians, look at the stats, and then stop the experiment. Okay, in this case it looks like the value was about 15%, which means this is a spillover value, or this is a compensation value that I applied to bring my population back to its original place. Okay, this is what happens when you run FITC only. I have not still shown how PE only looks like to the cytometer. Let's run PE only tube. PE was supposed to be in this house, but however it looks like it is slightly tilted towards the right. Now what does that mean? It means it is traveling towards your x-axis, which is FITC here. Although the travel is not so much, it still looks like it is sitting in its house. You have to look at the medians. If the medians are not matching, which means it's not correctly compensated. Now I bring this population back. Look at how the line changes now. It was slightly tilted. In this example, it is straight line. When the median of this population in A1 matches with the median of the population in A3, that's when I know I have correctly compensated. Looks like 400 and 420, more or less same. So that's how I know my compensation is correct. Remember, on a log scale, don't try to match it exactly word to word or number to number. 400 means 400 now. If it is 410, 420, 430, it is still okay, because these two are very insignificant differences on a log scale. Now, what was the compensation percentage I had to apply to bring this population slightly straight? Was 1.2%. Why does this happen? Remember, PE cannot spill too much back into FITC because the tailing is always more on the right side. Okay, perhaps PE will spill heavily into the next detector, not backwards. Okay, but I still have to do compensation, and that's the formula. The question again you'll have to ask yourself is, where is the spillover coming from? In this case, it's coming from PE. Where is it going to? It's going to FITC. The x-axis is FITC, if you remember. So if it is going to FITC, the problem is happening in FITC. So in the equation, FITC will sit first, minus a portion of this PE signal that was overlapping. That's the formula. Okay. Now, I have shown the detector how unstained looks like, which means without any reagents. I have shown how a single FITC looks like, which means only FITC alone, green alone. And then I have shown how PE, orange alone, looks like to the cytometer. Now the challenge is, if I mix all of them, because that's what I do in my actual sample, and throw it at the flow cytometer, will this be distinguished? The answer is a yes. If there was no compensation, this population in Q3 and this population in Q1 would have been in Q4. Why? because some of the FITC is also seen in PE, which is on y-axis. Some of the PE is also will be seen in FITC, which is the x-axis. So they will all appear in double positive, when in reality they are different populations. Why? Because that was uncompensated. Now what I'm showing you right now is compensated data of that of CD4 and CD8. Remember, CD4 and CD8 are mutually exclusive antigens, which means they are not expressed on the same cell type. If I have a T cell expressing 4, I call it as a helper type, and if I have a T cell expressing 8, I call that as cytotoxic type. How do I know if my compensation is correct? Not by visualizing it or by eyeballing it as we call, but by only way of comparing the medians. Looks like my medians are good enough here. Okay, these values are wrong, but then just look at the medians of Q3 and Q2, Q1 and Q2. If they are more or less same, that's when you know your compensation is correct. Now, just as an example, some bit of PE on the x-axis was flowing into ECD, which is Texas red, okay, another fluorochrome. So this was seen here. Now I have got it back, okay. Now I kept doing it, looks like it is going further down and it's in 0. Now what is this? Is this correct compensation? Is this overcompensation, or is this undercompensation? Let me help you with this one, and then I'll open up for the next question to all of you. Look at the median values of A plus minus, the Y median. It says minus 2.5. I'll read it for you. It's minus 2.5, okay? And I have to match this median with the negative population here in the first quadrant, A minus minus. A minus minus, the median value is 0.4. So you have 0.4 and minus 2.5, which means it is further down 0, which means I'm overcompensating it. I overdid it. So slightly get it back and match, let's say 0.3 and 0.4, and then you know you have correctly compensated. This is what I mean by median alignment. Now this is doable. This is good if you have, let's say, even 4 colors or even 5 colors, because you have to do all combinations. But then the moment it crosses 5 colors, you have to do something called auto compensation. So a cytometer that you buy must come with this feature of auto compensation. Most cytometers in today's times do come with that feature when you're doing with more than 5. Now, one of you or some of you can mute one by one, or perhaps whoever knows the answer, and let me understand the first case, CD4 APC versus APC A700, 2 different fluorochromes again. Some of the CD4 that was here was spilling into the double quadrant. I got it back. Help me understand whether this is correct compensation or not. One of you can unmute and let me know. Hello? Am I audible? Help me understand whether this is correct compensation or not. The first plot number A. The story is some of the CD4 APC was spilling into APC A700. I got it back, placed it here. How do I know whether this is correctly compensated? That's the question. Can somebody help me with this? Hello? Is it that difficult? Let me help you then. Okay. Let's take a look at medians now. The answer should have been, I will not know. I mean, visually it looks like the first one is overcompensated, but I do not know because I don't have the median values. That should have been your answer. But I'll give it to you on this one. Take a look at the median values of unstained. The unstained value sitting here is 2300. What's the value of the population that you just brought down? Minus 5000 indicates what? Which means your center ball, your negative ball was here, your negative mass was here. You got your population all the way down too much, which means you have overdone it. So you have to bring it back now and then match the medians. Okay. So the first case is really overcompensation. Take a look at B. Is it correctly compensated? Looks like, because the value of the negative is 2300, the value of the population that I just got back is 2290, more or less same,
Sir,
which means that is correctly yes, please.
Just one doubt. Yeah, actually, I had the same point. Like, we did not know the initial median value, but how did you calculate this 2300 one? So, is it from the
How did I
like?
calculate? This is from the
Yeah.
software. All softwares will give you this.
No. You just showed us the graph wherein 2.309% is being shown. So, how did you come on to the conclusion that is it 2300 initially? Because 5000 we could see minus 5000, but how did you see it was initially 2300 unstained one?
No. Unstained is what you see in this quadrant, right? This is what you measure from the software. Like this is the Y median of the unstained population. That's 2,300. I am still not touching my actual population. My actual population was somewhere in the dual region, which I know is wrong. I have to get it back. So the moment I get it back from here, I kept doing it and I overdid this. So it went all the way down the negative scale. So that's now 5,000 minus 5,000. Okay. So the only reference I have is 2,300 and my 5,000 is here. Bring it back and align it with 2,300. Did you get my point now?
Yes, sir. Yes, sir. Sir, with the B and C part, that means they are like correctly compensated because other things.
No. To take a look at the value of the B. What do you think?
Sir,
2,300 and
it is
2,290.
negative. It is negative, so it is overcompensated.
No. Take a look at B plot again. Look at the unstained value is 2,300 and the
Yes.
population that you just adjusted is 2,290. Are they close to each other?
Yes.
Are they correctly compensated? Looks like yes. Okay. And visually also it looks like it is. But the only way I confirm is using medians. Take a look at plot C now. Your unstained value is
Much lesser.
2,300. Your actual population is,
7,000.
which means it is still lying up
Yes.
on the positive y-axis. So you have to still get it down. There is still a lot to be done. It is under compensation. Okay.
Yes.
So the first case was overcompensated. The second was correctly compensated. The third was undercompensated. When your population value is higher than unstained, that's undercompensation. When your actual population value is much lower than unstained, that's overcompensation. When they match with each other, that's when you know it is correct compensation. Okay. Right. Now, sometimes when you do this compensation, the problem is some of the cells will stick onto the axis. Okay. Now, this is what happens when you do compensation. So most modern day cytometers will give you something called a bi-exponential scaling. If you see the arrows that I've indicated here, they're all some populations in pink, noted as pink. Is my screen presenting still?
Yes,
Somebody
sir.
can.
Yes,
Yeah,
sir.
because some Amol Salve was trying to present it. Okay.
It's there.
Now, thank you. Now, all the populations are sticking inside the axis. So I don't know if they are really negative values or if they are my actual populations, or am I missing them? Am I, not counting them? So the only way to know that is use something called bi-exponential scaling, or on some software it's called logical scaling, some it's called TrueView. Simply put, bi-exponential scaling means introduction of a linear scale to the existing log scale. So if you have log scale, let's say 10 power 1, 2, 3, 4, at the lower end of the axis, below 10 power 1, you start introducing linear scale, 1, 2, 3, 4, all the way to 10. Okay, that's when whatever was sticking outside, inside, will now come out. You see the difference between first and the second plot. No difference absolutely, it's just a better visualization of plot A. This is what I mean by exponential scaling. Having log at the high end and linear at the near the axis is what I mean by X scaling. Okay, why does this happen? Because on a log scale, small values, for instance 1, 2, 3 or 0.1, 0.2, 0.3, cannot really be distinguished properly and they appear in the large negative region. To pull them up and do a better visualization, you have to use by exponential scaling. Some softwares in today's times, like the Site Expert on the Cytoflex, is default by exponential scaling. So you will always see the population this way, never this way. Okay, so that's the progression of flow cytometer. Now, we talked about compensation because when it comes to immunophenotyping, you're trying to match antigens with different fluorochromes and study a single sample, let's say a 50 microliter of blood sample for your immunology experiments. That's the reason why I taught you what should be done before you actually begin your experiment, something called spectral overlap and compensation. The application of immunophenotyping, you can do as many as experiments as you can. For hence, apoptosis, you can look at the cytokines level. For instance, in case of COVID-19 now, you can look at the immune response, you can look at classifying different phenotypes, whether you have T cells being recruited, B cells being recruited, and if yes, how many, if yes, are they functional, what cytokines do they release, etc. And you can also look at phosphorylation, stem cell biology. All of these are possible if you learn the basic tenets of immunophenotyping well. What is essentially immunophenotyping? You're looking at expression of proteins on cell surface or inside a cell, which is cytoplasm, and then you're trying to identify a particular cell based on that protein. It's like a unique signature for that particular cell type. Okay. Now you can also just simply say, okay, I have a population or I don't have a population. Or if you have a population, you can also study how many number of those antigens are present on my cells, which means if I am trying to study CD4, I can also please mute.
But we have issue that we have to also see that we are producing.
Hello. Yeah. Now better. If you are only looking to classify, which is a quantal assay, as we say, or a qualitative assay, yes or no answer, whether the population is present or the population is absent, it's okay. But then you can also quantify, meaning how many number of my CD4 antigens are present on my cell surface. Okay, you can also determine that receptor count on your cell surface. That's also the objective of immunophenotyping. Like Professor Kumaresan pointed out in his session, you can use various techniques available in biology in today's time. You can do a Western blot, you can do an ELISA, you can do immunoprecipitation. The advantage with flow is you're looking it at on a cell to cell basis and not an aggregate information. Whereas in ELISA or in a Western blot, you're just looking at an aggregate information of the entire cell lysate. 10,000 cell lysates put together will give you some information and indicate something. Whereas here you're talking to a cell to cell basis. That's the advantage of immunophenotyping on flow. What can I use immunophenotyping for? For example, the most obvious answer would be you will look to study immune system, whether it is innate biology or adaptive biology. For instance, if there is an infection like the one we are facing right now, COVID-19, the first sign of immunity would be innate, whereas your macrophages or a specialized form of macrophage called dendritic cells will pick up some protein of your invading pathogen and then...
Mukund, this is Babu. Yeah, sorry to interrupt.
Yeah.
His screen is not coming up again for
Is it for everyone?
Yeah, it's for everyone.
Sir, it's
Can you just present again? So the
Okay, I will do it again. It looks like it could be because of it. Okay, see, that's the problem. Babu, perhaps it could be because of internet connectivity. Some will get it.
no, not from my side. From the other side also.
Okay, I'm sorry about that. Now I think it should be back.
Yeah, now it's
so
back. Now it's back. Yeah, now
thanks,
it's back.
Babu. Right. Now, what I was trying to talk you through about is the immune system studies itself using immunophenotyping. Of course, when you have an invading pathogen, the first sign of the first sort of cells who will kick in is macrophages, dendritic cells will pick up a portion of the protein of the pathogen, and then they will go and talk to their friends, which is essentially the T helper lymphocytes, present this antigen to those, and then they in turn start identifying this antigen and then start talking to all other friends in the immune system and starts recruiting them. Okay, this is the chain of events whenever there is an invasion or a foreign invasion in the immune system. Now, how do I study all of these things using, let's say, just 100 microliters of blood sample or one swab of perhaps saliva? How do I know whether I can study all of these things? Remember, each of these molecules or each of these cells, rather, will present or have a different antigen receptor on their cell surface or within the cytoplasm, or as in when they start traveling, when the T lymphocytes becomes activated, it will produce a different protein altogether. It will have CD25 in its activation stage, whereas in its resting stage, when it is not doing anything, it won't have CD25. So differential expression of antigens and presence or absence of antigens as they mature into different cell types will help you do immunophenotyping. I can tag different fluorochromes to all these antigens and study immune system in just about 50 to 100 microliter of sample. You can study as many as 20 antigens. That's the power of flow cytometry. And all this, remember, at about 100,000 cells per second. That's the rapidity. What do I mean by these molecules that I talked about? These are essentially called as cluster of differentiation, or in other words, CD antigens. CD is cluster of differentiations. As we speak now, I think we have about roughly about 380, 390 CD antigens being classified for humans alone. Likewise, you have some for mice and other species as well. If you look at the example that I've shown here, a T helper and a T cytotoxic cell, a T helper cell will have all these antigens expressed on its cell surface. The signature molecule among all of these things is CD4. So if a particular T lymphocyte expresses CD4, I know for sure this is a helper type. And likewise, if it expresses CD8, I know for sure it is a CD type, a T cytotoxic cell type. Okay. You can also study different stages of differentiation or maturation or activation. Like I pointed out, for instance, if you have an immature cell like CD34, which has the capacity to become any cell type in the body. Now, a CD34 expressing stem cell will progress, will differentiate itself into, let's say, a monocyte, a granulocyte, a lymphocyte, all the way from this lymphocyte becoming activated and becoming an activated lymphocytes when it expresses 25. So if you notice carefully, CD34 is gone when they become mature, okay, which means the immature marker is now lost. They have acquired new markers. So I can really study how these cells are differentiating into different cell types basis the marker that they express. Okay, this is how immunophenotyping can also be helpful for all of us. Two standard methods of immunofluorescence using flow, in fact most other techniques as well, is imagine this is a cell and the red triangles that I indicated here are antigens. An antibody binds. This antibody is directly conjugated to the fluorochrome indicated by a light bulb here. Okay, I repeat, you have a cell, you have antigens, an antibody bound to a fluorochrome binds to it, and then you incubate them further, wash it, analyze it. Okay, this is a direct immunofluorescence where you're using a directly conjugated commercially available antibody to do your studies. For example, let's say some of you are drug researchers and you have developed your own therapeutic monoclonal antibody against cancer or against some viral infection, for instance. How do I do then? Imagine this is your antigens that you're targeting. These are your targets. This is your antibody that you have developed, raised in mouse but anti-human. Okay, so this is the primary antibody. To this antibody, I raise a secondary antibody. Okay, the first antibody was raised in mouse. Now I have to raise a secondary antibody anti-mouse in other species. Okay, so the secondary antibody is bound to fluorochrome. That will in turn bind to primary. Primary is in turn bound to antigens. So this is an indirect way of assessing immunofluorescence. What you primarily do is label cells with primary, incubate them, let the antibody and antigen interact with each other, wash, get rid of excess antibody, again incubate them with the second step of secondary antibody carrying the conjugate, wash, acquire, and analyze. So these are the classical methods people use for flow cytometry immunofluorescence studies. When do I use indirect? For examples, when I told you when you don't have the commercially available conjugated antibody for your area of work, or let's say you have your own antibody in the lab where you want to study that, that's when indirect way of immunofluorescence using flow will help you. I have already shown you this slide. This just indicates what are some of the different fluorochromes that I can have in flow cytometry from different lasers. Remember, there are about 10 fluorochromes here, and I can tag all these 10 fluorochromes to 10 different antigens. Using 50 microliters of blood, I can study all those antigens all at once within 5 minutes. That's the power of flow. That's the power of immunophenotyping flow cytometry. How do I read a signal? I'll repeat this again for all of you. If you have very little antigens, obviously very few antibody fluorochrome combination bound, you have a low fluorescence signal. If you keep adding more and more antigens, if there are more and more antigen and antibody interactions, the signal will start increasing from left to right. That's how you read a flow cytometry plot. How do you read a dot plot? Some of you asked me, how do I classify different cell types? This is one good example for you again. On the x-axis, you have CD4 FITC. On the y-axis, you have CD8 PE. Okay. Now, this is a double positive, which means it's a double negative. I'm sorry. This means it is negative for CD4. It is negative for CD8. Okay, which means this is not expressing either 4 or CD8. So in a lymphocyte, what are some of the cells that we don't express 4 and 8 both? It could be your NK cells. It could be your B cells. Okay, all of them are lying here. The moment I add a marker which is CD4, I know for sure these are my T helper cells because they are expressing CD4. Is the expression bright enough? The answer is no, because it sees it is just one log away from the negative. It could be dim expression of CD4. Okay, perhaps because you didn't add enough antibody, or perhaps there were not much antigens available on that particular cell type when you actually did the experiment. Likewise, you will also see a comet-like appearance of CD8 population. This is the standard appearance of CD8. You will have a head space where you have most of these dots accumulating, and then you'll have a faint dot like a tail. Okay, this looks like a comet tail. This is a classical appearance of cytotoxic CD8 positive cells. Okay, look at the expression. If this line is negative, looks like there are some which are dim positive, like here, and there are some CD8 populations which are very, very moderate. Okay, so this is heterogeneous expression. This is what I mean by heterogeneous. Now, the first plot that I just talked about is exclusive antigens, which means these two antigens, 4 and 8, are never expressed on the same cell type. Take a look at co-expression on the second plot. You have a CD4 on the x-axis and a CD3 on the y-axis. Okay, this double positive region indicates expression for X, which is CD4, and expression for Y, which is CD3. And what are these cells which are expressing both CD3 and CD4? These are again T helper types. Okay, it's just that instead of CD8, I have added CD3 marker here, and all the population moved to, from here, move to the double quadrant. This is a quadrant of dual expression or double expression, or simply put, co-expression. Okay, this is how you simply distinguish different types of cells based on different markers. A simple protocol for all of you, like I told you initially as well, take antibody, for instance, anti-CD4 and anti-CD8, add them to the blood sample, for example, 50 microliters. Because you've added antibody and the antigens are present on blood sample, allow them to incubate for about 20 to 30 minutes, usually 20 minutes, in room temperature, dark conditions. Why dark? Because you're dealing with fluorescence. If you do it under bright light, the fluorescence will be quenched. Quenched means the fluorescence is already gone. So therefore, incubate in dark. And then because you don't want RBCs, add a RBC lysing agent, commercially available buffers or an ammonium chloride based buffer. Allow the RBC buffer to act. Let it sit for 10 minutes. Again, room temperature, dark conditions. Because there'll be a lot of dead cells, a lot of debris in your sample. Get rid of excess debris and get rid of excess unbound antibody. Spin it down. Remove the supernatant. To the pellet, add PBS buffer. Add maybe a fixative of 0.1% paraformaldehyde if you don't want to analyze it, let's say, within the next 1 or 2 hours. If you anticipate that the facility will be free only, let's say, in the evening hours, add a fixative, run through the cytometer. This is called a stain, lyse, wash protocol. Why stain, lyse, wash? Because you're staining with an antibody, you're washing to get rid of excess antibody and debris, and then you're also lysing the RBCs. Okay, this is how a simple human blood immunophenotyping experiment can be done. There's one more protocol called lyse no wash. This is what people follow in the clinic. When I say clinic, in the hospital side, on the diagnostic side. If, for instance, if you're trying to enumerate the number of CD4 counts in an HIV patient, okay, you're trying to do immune monitoring studies and trying to assess how many number of CD4 cells are available in a HIV patient, you cannot wash. Why? Because if you wash, every time you lose a lot of cells. And if you lose a lot of cells, you're not knowing the exact number of CD4 cells in an HIV setting. That's why you don't wash. But when you don't wash, you will have, remember, a lot of debris, a lot of contamination from RBC, a lot of broken cells, and etc. So whenever you use a no wash protocol, you have to use CD45 antigen, or you have to use CD45 antibody. Why? Because I am trying to study CD4, which is a WBC, and if I add a CD45 antibody, I can separate WBC from RBCs now. So the contamination question is gone. Then I can study different subpopulations of lymphocytes. So use of CD45 is a must when you do a lyse no wash protocol. Now question is, how do I count? Okay. Remember, flow cytometer cannot tell you exactly when you pass through 100 cells, 100 cells will what will be recovered eventually, because there will be something called aborts, something called coincidence. We'll never recover 100 cells, which means it's a relative science. How do I make it absolute then? Take a reference. I pass 100 number of beads and I recover 95 beads. Okay. So naturally, in the same system, same settings, if I pass hundreds of cells, if I recover, let's say 95 or 96 cells, assuming 1:1 ratio, that's the absolute count. So you have some bead reference to do the measurement of your count. Okay. Usually it is a 1:1 ratio. If I add 50 microliters of beads, I add 50 microliters of my blood and then do the counting. Okay. This is how you do something called absolute counting using a reference bead method on a flow cytometer, but you have to have a CD45 marker in the sample when you are doing a no wash, because you're counting cells. The formula is the number of cell events that you get from the software, the number of the bead events, how many recovered, basically how many did you recover in terms of cells, how many did you recover in terms of beads. Okay, this is the statistical output from the software into the total number of beads that were available in the beads itself. When I added 50 microliter of bead, as a manufacturer, I'll give you or I'll tell you what does 1 microliter of my bead have. Okay, it might have 1000 beads, it might have 2000 beads. I really don't know what 1 microliter of that has. So that's the total count divided by what's your volume. You will get your absolute count. This is mostly followed in the clinical science when they do, let's say, hematopoietic stem cell CD34 counting for transplantation and CD4 when it comes to HIV immune monitoring. The last couple of slides, in fact, I think this is 1 or 2 slides before I end, is we talked about a lot about surface staining. How do I do intracellular staining? I'll just leave you with a teaser of how do you do intracellular staining. Imagine you have a stimulus, which means you have some stimulant which is going to induce some changes downstream of your cell. Okay, let's say you are using a plant lectin and trying to study the anti-cancerous properties of this. I add lectin as a stimulus. I induce activation on my cell types and then stain it with different antibodies. For example, if I'm looking at effect of lectins on lymphocytes, I induce activation and then see whether my lymphocytes are actually activated or not, or if they are not, what else is happening. So I will study CD4, I'll study CD8, I'll study CD25, of course CD45, etc., and then add antibodies, and antigen and antibody will interact with each other, analyze on a fax machine. Okay, you can study the effects of different stimulants on the same cell type or same stimulant on different cell types in intracellular staining. People also use this exclusively or extensively in the clinical flow cytometry when they talk about diagnosing cancers of the blood, which is leukemia and lymphoma. When they look at, for instance, myeloperoxidase, MPO, and they also look at the TDT nuclear antigen for diagnosing some of those leukemia conditions. Okay, so this is the simple way how you do an intracellular staining protocol. Now, how do I club all these together? Is it really happening? Whatever I just told you, does it really happen? The answer is yes. These are some of the commercially available antigens from Beckman Coulter. We call them as Duraclone because these are room temperature stable. If you looked at first tube, these are all pre-titered, pre-cocktailed, all dried in a 5 ml tube. All that you have to do is add blood, lyse the RBCs, and use them straight away for your analysis. Okay, the first tube, if you look at, you have 45, 16, which is a granulocytic marker, and then also an NK marker, CD56, a natural kill cell marker, CD19, a B cell marker, CD14 is a monocyte marker, 4 is a helper T, 8 is a cytotoxic T, 3 is a T lymphocyte. So I'm doing basically T, B, NK biology using just one tube, 50 microliter of samples straight away. So whatever I spoke of is actually not imaginary, it is real. These are commercially available. Likewise, if you want to do B cell or a dendritic cell or a Treg tube, depends on your experiment, depends on your hypothesis. This is just a simple, ready to use panel. Okay, I will talk about this and end my slide and take questions perhaps. A lot of publications that came out in the last, perhaps let's say 15 years or even 10 to 15 years, there was no real standards for reporting flow cytometry data. Some people might include the specimen type and the time of collection, some may not. Okay, there was really no standardization when it comes to reporting standards. Okay, so what ISAC and this community of other biology societies did was to come up with a standard called flow cyte, which is the minimum information about a flow cytometry experiment you have to provide if you want to get accepted to a good journal. Okay, this is a good practice, the best practice for data publications, especially for all the researchers out there on call today. Please read this article. I'll not try to give you a lot of details as to what information is required before you publish. It's a fairly simple 2 to 3 page article, and this is an open access article from the National Institute of Health with the same name, flow cyte, the minimum information about a flow cytometry experiment. What they basically ask is, what is the goal of your experiment? Why did you do this experiment? What are some of the variables? Are you inducing any changes? Are you using a stimulant? What's your conclusion? What's the quality control? What are some of the controls that you used? What was the sample type? How did you analyze the data? What is the instrument you have? What is the optical layout? How many detectors? How many lasers? Etc. 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 So when you submit this data to this flow repository, a lot of people sitting across the world, I mean different cities, different places, will know your entire experiment in and out. They will then be able to do a better diagnosis, a better review of your data. That's the whole purpose of MIFO site guidelines. Okay, so that's it from my side in terms of immunophenotyping and flow guidelines. I open it up for questions. We know we have overshot it by 5 minutes. I'm sorry about that. I open it for questions right now. If you want to read, so these are the best book for reading. If some of you wants to do a basic reading, I suggest you read this book by Michael Ormerod. This is available online, accessible as well, flowbook.denovosoftware.com. Dr. Michael Ormerod, basics and a lot of applications covered in great detail. If you want to spend the rest of your career using flow cytometry, or if you're an immunologist, I recommend highly reading this book. This runs into about 600 pages, so pace it, read it at your leisure, but then it's a very, very good book. Thank you so much. Open for questions.
Sir, I have a question.
Yes, ma'am.
Before that, I have to tell you that it was very informative, very nice and lucid presentation. My doubt is about the compensation slide where you asked us to unmute. Can you please go to that slide, sir?
Yes, please. This one?
No, sir. No. Yeah, this one. Let's practice. Here, there are both the stained, no, sir? Both are stained, like APC, CD4 and CD8. Dual staining can be performed compensation, or a single stained cell, like a sample. Are you getting my question? I could see both are stained here, like CD4 and CD8.
Yes.
You were saying that
your question is, can I, if I have some population here in the dual positive zone?
Not the dual positive. I'm asking about the CD8 APC A700A. So there is population there.
Yeah. Correct. So what's the question?
So dual staining. This is a tube of dual staining, CD4 and CD8. So compensation can be performed on a dual staining cell sample, or only a single stain sample we have to perform first compensation and then.
All right, I get your question. See, if you have mutually exclusive antigens, okay,
yes,
which means in case of CD4 and CD8, don't do any tube 1, tube 2 or unstained. Straight away take the mixed tube, because remember, even this tube will have unstained cells, right? Like I see here, correct?
Yes.
Use the medians from those. And because these are mutually exclusive, one will be here, the other will populate there. So there will be no double positives. So you absolutely know, because the biology is as such, I can still use one tube and do my compensation. If you are only working with two antigens, CD4 and CD8, and mutually exclusive antigens, okay?
Yes, mutually exclusive. I got
mutually
it. Mutually
exclusive,
now,
you can do.
Yeah, but you are saying CD4, you have moved it down, right?
Yeah. This is overcompensated.
There can be cells which can be CD8 positive, but you are moving it down forcefully. Is it possible?
No, because you don't have any CD8 positive here. Why? Because these are mutually exclusive antigens. CD8 will always be here. I'm not doing anything.
Show some spillover like that.
This
thought.
one, these,
Yeah.
no, this
Feeling
is all.
hate towards,
This is all the same population, just bringing it down. Some of the reasons why you will also still see, despite being mutually exclusive, still see
yes,
some
sir.
in the
That's
dual
what.
quadrant is because of something called doublets. These are aggregates, random
Okay.
order aggregates. These are basically artifacts in your flow. Tomorrow, when I talk about this in doublet discrimination, I'll tell you how to distinguish this as well. Okay.
Okay, sir. Thank
But
you
your
so
second
much.
question of can I do this on a co-expression? The answer is no. You have to use single color.
Okay, sir. Thank you, sir.
Thank you, ma'am. Are there any questions in the chat box, or have I bored you too much? Won't work until it's changed to public. Okay. Why tailing is more right side as in case of PE and FITC? That's the property of the fluorochrome itself, Smritishri. I mean, any fluorochrome that's naturally available right now, the tailing effect is more on the right side. I mean, it can't go back the other ways. The only way you will get rid of this contamination or spillover is by compensation. If our study requires to identify inception of PE curve, what do you mean inception of PE curve? See, if you're trying to measure only the excitation spectra of PE, then you can do fluorimetry. I mean, if you're developing your own conjugation in the lab, there are kits available for that. If you have your own antibody, a separate conjugation, mix them together and try to measure the emission and the excitation maxima, you can use fluorimetry. But then if you're trying to do flow cytometry, you will have spillover issue if they are spectrally close to each other. The only way to correct that is compensation. Thank you for all your wishes. I see people have liked it. Please. Cell sorting. Okay. What do you want to know about cell sorting? I can talk all day about cell sorting. Cell sorting essentially is the first bit of that is analysis. Basically, whatever we taught in the last 2 hours. And then you apply charge on the different populations, for example, CD4 and CD8, send them to different collection vesicles, purify and use them for further your genomic applications. That's what sorting means. Sorting essentially is like the purification of cells. What you do with respect to HPLC when it comes to protein is what we do with cells when it comes to FACS. It's basically cell purification like your HPLC protein purification. Okay. The method is charging. There are also magnetic methods also. Some say voice not clear. I am sorry, I didn't notice. The form issues, I think, sir, will take care of it. Don't worry. Are there any questions, or you're too hungry? We'll let you go.
Sorry, sir. I have one more question.
Yes, please.
You haven't talked anything about the antibody concentration, how much we have to use before setting a new experiment. Like, there is a process, no? Like antibody
titration.
concentration. Yeah, titration.
I'll talk about that tomorrow, ma'am. That's why I didn't do it today.
Okay, sir.
When we do multicolor, one of the steps and one of the important steps is antibody titration.
Yes, sir.
And knowing something called stain index, I'll talk about it tomorrow. Don't worry.
Okay, sir. Thank you, sir.
So, Moon sir, thank you very much, sir. It was a fantastic and a very clear lecture, and I think we could understand the principles very clearly. Thanks a lot. It's wonderful, and we will close the session. And all of you guys, please come back. We'll rejoin at 2:30 p.m. with the lecture of Dr. Rajendran on applications in microbial aspects. Okay, sir. So we'll resume viewers tomorrow. I think 11 a.m.
Sure, sir.
Okay, sir. Thank you, sir. Thank you very much, sir. Thanks a lot.
Thank you. Take care, everyone.
Thank you, sir.
जल्दी पर ಖಂಡಿತಲೆ ಕೊಟ್ಟಿರೇನ್ ಬಾಬಾ ಬಂಗಾರಿ ಓಮ ಒಳಗಿಡ